Hello again, everyone. Welcome back and good morning. Welcome to the our last day conference. Uh, I hope that you are still, some kind of energy to end the day. Hopefully, I see, happy faces. We're going to start our session 8, which is data standards and ontology. So it's my honor, a pleasure to present our speaker, Dr. Monica Munoz Torres. So Dr. Munoz Torres is an associate professor of biomedical informatics at the University of Colorado Anschutz. Her works focuses on critical challenge of developing the socio-technical foundation need to realize the promise of artificial intelligence in biomedical science. She leads the NIH-funded Bridge to Artificial Intelligence, Bridge to AI, teams focusing on standards, practices, and quality assessment, as well as teaming and collaboration. Dr. Munoz Torres also co-leads the clinical and phenotypic data capture workstream in the Global Alliance of Genomics and Health, GA4GH. So one of the quotes from Dr. Munoz Torres is, to build trustworthy AI, we must first establish trustworthy data. So Dr. Munoz Torres' work centers on defining the character of the data. So the talk will be entitled Bridging the Gap, Foundational Criteria and Standards for AI Ready Biomedical Data. So sorry, I didn't present Henry, who will be co-sharing the session with me. So welcome, Dr. Monica Munoz Torres. The floor is yours.
Good morning, everyone. How are we feeling last day? Lots of good energy, lots of good dancing last night. Yeah. Welcome back to the science. So I hope that we're going to spend the next 19 minutes and so talking about two things that I'm very passionate about, which is organizing data and also artificial intelligence. And so I entitled this talk "Bridging the Gap," as it says here, foundational criteria and standards for AI-ready biomedical data. Very long, but you can see over there in the corner. It should be called something like From Curation to AI Readiness. So why don't we go with that title? Thank you so much for being here. I'll take you on a journey introducing you to my project. Me and 400 of my closest friends work on the bridge to artificial intelligence. I'll tell you a little bit about the conversation that we have been having over the last few days on how biocuration is evolving. I'll tell you about the requirements for AI readiness and how we developed some criteria and practices for biomedical data and how this impacts our community going forward. The Bridge to Artificial Intelligence is a project that funds approximately $130 million of the United States for coming from the National Institutes of Health. And it has three basic pillars, one dedicated to the distinctive, fair, and AI-ready data sets, trying to propel modern artificial intelligence and machine learning models. We also focus on ethics, making sure that the data sourcing happens within the principles of, having a culture of ethical consideration for their generation. And we also focus on the people, not just the people that that the data is being generated about, but also who is generating those data. And so this is an intention to try to modernize the workforce and making sure that they are skilled in this new methods of data creation. Bridge2AI.org, it's our website. There are 4 grand challenges at the National Institutes of Health in the United States that we're trying to address. The first one is called Precision Public Health. In this project, we're collecting data for approximately 10,000 distinctive bioacoustic waveforms. So this is about collecting the speech for studies in mental health, respiratory, neurological, and other areas. There's another data set being created for the Salutogenesis project. This is about the 4,000 individuals around the pathogenesis and cytogenesis to expand applications in artificial intelligence. And this focuses on type 2 diabetes patients. And then there's a third clinical project centered on the collection of data from intensive care units across 14 hospitals with approximately 100,000 patients. So millions of rows of information coming together. And as you can see on the right-hand side, there's a lot of very specific clinical data being captured for this. And you can see the heterogeneity. So it's multimodal, is emergent. We have things like, an ECG machine and information from a patient in an ICU unit, all the way to glucometry for patients with diabetes. Our fourth project is called Functional Genomics and focuses on generating cell maps, perturbing about 200 genes across the human genome in these cell lines, and then using immunofluorescence imaging, mass spectrometry, and transcriptional profiling to generate these maps. Been working together for approximately 4 years. And you and I, working with our data sets, have come to the realization that there are several challenges and opportunities on how we organize this data. And so we know that data sets must be rigorously curated in order to achieve accuracy and completeness and consistency, and that poor data will lead to poor models of artificial intelligence. We also know that we have to have very transparent data practices so that we can achieve, better explainability for the artificial intelligence models, and we can enable users to both understand and trust the insights that we're deriving from these models. And last but not least, we understand that the applications have to address ethical concerns through documented data provenance. All of this trying to get to ensure that all of us curators and the broader scientific community has the ability to ensure responsible and fair biomedical research from these data sets. I think this theme started a few days ago, and so what I'm about to share, you're gonna go, yeah, Moni, you already said that. We have seen that traditionally, biocuration has been focusing on managing these human readable knowledge bases as annotations, literature, and metadata. And this rise in artificial intelligence demands has turned our field into a field that necessitates the creation of AI-ready data sets so that machines can actually interpret what we're generating and putting out there. This involves better structures for the data, better metadata, better standardization formats, and making sure that our computational workflows also evolve with the evolution of the data. You and I now become engineers of the data sets to try to support these machine learning models, and we thereby enhance the reproducibility and the transparency of our research products. So Alex totally stole this punchline, so I'm gonna just repeat it because AI becomes then a complement to the expert curator and leverages high quality standardized data to accelerate discovery. The question of how do we best utilize and leverage all of these AI technologies to improve the biomedical data that we collect and curate continues to evolve. What I say today will likely be different in about 6 months when the technology changes. And we just have to remember that the data sets that we generate and curate alone are not ground truths. We need use cases and we need metadata to explain them. And so we've provided those criteria here. I'll introduce you to the AI readiness criteria based on the principles of the Bridge to Artificial Intelligence project. They are generalizable, so they will, I hope, apply to all of your data as well. We start where we all know, fairness, right? This is ground zero, and that's why we call it zero, because all data sets should be fair. We know that one by heart. But we also need to have information about origins and transformational history of every digital object. And we also need to have very robust and thorough characterization of these digital objects, semantics, statistics, standardization properties, and we want to describe all of these data sets in software, including quality and biases. We then move on to pre-model explainability. While it shows up here in number 3 in that yellow bit, okay, maybe it doesn't. There we go. Well, it shows down here on pre-model explainability. It actually should be around this entire wheel. For those of you online, I'm just making circles around the AI readiness criteria wheel because Supporting the explainability, the data that we generate should be supporting explainability of the models and the predictions and classifications based on the data with regard to the metadata, its fitness for purpose, and the integrity. And this should actually be something that we take into consideration at every step of the data lifecycle. We also highlight that we must have considerations for the ethical acquisition of the data, its management, its dissemination, and how it is maintained and documented. And in addition to these ethical principles, we should think about how the data are then going to be stored long-term and archived. So thinking about sustainability from the moment of inception is critical to make our data AI ready. And last but not least, this is the Captain Obvious moment, of course. We want to make sure that all of the data are standardized, computationally accessible, portable, and contextualized so that there is interoperability and ease of reuse. So keep this wheel in your back pocket from now on. I hope that you share some feedback with me about which of these aspects I may not have considered that you think are important. This is what we did. We actually created a worksheet so that anyone can self-evaluate their data sets against this criteria. In this worksheet, we have divided all of the criteria that I just listed. In this particular one, we have provenance as an example, and some of the elements that we assess in order to look at provenance as whether the data sources are traceable, so transparency. Whether we can identify and transform all of the transformation steps, whether we have interpretable information, if the software for key data transformation analysis are available for the repository, and who are the people and organizations responsible for and processing the data, the samples, and everything related to our data sets. So we've done very thorough explanations for each of these criteria. And then we have actually given these data sheets to our own projects. So our 4 projects, as you see, the functional genomics, artificial intelligence for clinical care, precision public health, and salutogenesis, all self-evaluated. And we realized that there are some opportunities and some challenges. What you'll see, where you're looking at that is basically a radar plot where each criterion is treated as an axis in our radar plot. And so on the outside, you see oopsie daisies, sorry. On the outside, you see the orange, which is the goal of having 100%. And then in the inside, for those on you online, there are like blue lines, which are the current state of each of these grand challenges. The idea, of course, is to try to get to having the blue line and the orange line overlapping. And these radar plots help us visualize the AI readiness features that would benefit from have some improvements, discussion, and implementation. For each of these particular projects, for instance, on the top left on the functional genomics one, we see that they didn't really have information about the potential sources of bias, or some summary statistics were missing. So for that, they got dinged and they do not have the complete criteria. And then similarly for characterization on the salutogenesis. The goal is not, of course, to tell the project, nainer, you didn't do it, but rather help them figure out what are the best ways for them to achieve these level of AI readiness according to our criteria. And the worksheet helps them keep track of this. These are figures from a few years back, a year ago. And as of today, we're actually finalizing the evaluations with our grand challenges, and all but one has now achieved 100% across. And so it's been really good to work with them and provide them feedback for this. Why is this important, and why should anybody care? So I'm really, really confident, and I hope that you support me in sharing that the transparency, the characterization of the data sets really provides clear documentation of the origins of the data, as well as the methods for collection. How do we pre-process? How do we ensure transparency from acquisition through model training and deployment? So this is thinking about how this will be used downstream for artificial intelligence and machine learning. And then we also know that this characterization also helps us identify potential sources of bias. And this allows all of us curators to implement strategies that help us minimize the unfair or the misleading artificial intelligence outcomes. We also want to make sure that we have data sets that comply with ethical standards from the beginning. We have 100, at the moment, 90,000 patients in ICU. They're unconscious. They cannot consent to the collection of their data. But we have worked with IRB boards and university protocols and entities to making sure that the data are both sourced and treated ethically, so that the protection of persons is validated. And last but not least, if we work on better characterizing our data sets, then we're going to have better pre-model explainability for these models, and we're going to give better context to the data. We're going to have better integrity checks and make them better fit for purpose. So one way in which we actually have operationalized the representation of data about a data set is by, we have a few tools, and they're open source and they're available in some document to help us document the data sets. And we have leveraged a modeling language that we were discussing in one of the talks yesterday, LinkML, to be able to standardize information about the data sets. I'll skip to just say, this is called data sheets for data sets. If you remember in 2021, the Google group, Geber et al., came up with an initial schema that introduced this like milestone generic dataset schema for all datasets, not specifically focused on biomedical data. Over the last 3 years, we've been augmenting this and modifying it to expanding on its robustness to make sure that we can offer some comprehensive documentation of AI data sets, and we're enhancing transparency, reproducibility, and accountability for AI research by doing so. In here I have a few links so that you can go and explore what these data sheets look like. This is how we have implemented them. They're serialized both in LinkML schema, but also on Pydantic classes so that you can use them. They're based on schema.org vocabularies, and we have made sure that we have modules that now help us explain relevant biomedical research terms, as well as provenance ontologies. And this gives us flexibility for a more comprehensive representation of the metadata for your data sets. We utilize LLM assistants in agents in order to generate this data set. So we utilize them for the initial process of grabbing all of the available information, websites, documents, the research proposal for the data set, and grab it with the support of a large language model to organize it and then validate it against the schema that we generated. And then we also have on our repositories a little assistant that you can visit today and go ask it, hey, I have these little information about this data set. Would you help me create a data sheet? We also have the Standards Explorer, which is part of the suite of tools from the Bridge to Artificial Intelligence. And if you visit that website that I have placed down here at the bottom for those of you online, is the third column bottom website. You'll see our Standards Explorer, which gives you the opportunity to visit information about each of the grand challenges and look at what their data sheets look like right now. We're evolving these. We have our all hands meeting next week, so these will actually change sometime on Monday night. And last but not least, we have an integration with Fairscape, which is an environment that we're using to help us round trip user-annotator dialogue. So we have curators that we've enlisted from the projects, and we tell them, here's what the data sheet looks like. And this is all of the information that we captured about the project. Do you agree? Is this sufficient? And they have the ability to give us some feedback about this. All of these links are on the slides. I'm sure the conference is going to make these available. So as you can already tell, achieving AI readiness is not a one-person job. This has to be a collaborative task. There's a lot of necessities for a deep understanding of biomedical data. We need to understand predictive modeling tasks, scientific domain knowledge, statistical methods. And we also have to take into consideration that this concept of AI readiness is one thing today and will continue to evolve. And in a few months, it might be modified because there will be other technologies for artificial intelligence that change some of our practices. So we have to evolve with the technical aspects. And the ethical governance and the sustainable data stewardship are also foundational and helps us ensure that we responsibly use the data and give long-term accessibility to these. the impact that I see of these practices in the biocuration community is that I think with these criteria, we're offering a roadmap to help all of us in biocuration manage the increasing complexity of the data and help us ensure that our data sets meet more rigorous quality, provenance, and ethical criteria. We're also helping with giving you as curators the possibility to build this symbiotic relationship where the artificial intelligence tools are actually augmenting your curator's expertise. And we're also I truly believe that this is empowering to all of us in biocuration community because adopting standards like this will help us to making sure and shape more sustainable, transparent, and reproducible AI-driven biomedical research globally. So there are a few things that we're thinking about, as I was saying, making sure that we evolve these criteria as the technology evolves, as the data types evolve as well. There are new data all the time. We also want to make sure that we evolve these practices, emphasizing the integration of ethical principles and principles of sustainability. We want to make sure that the development and adoption of advanced open source tools can enable scalable, reproducible, and transparent AI workflows. And we're hoping to strengthen the collaborations in an interdisciplinary manner that brings together data science, domain expertise, and artificial intelligence expertise. I'm hoping that you will feel empowered because your work drives consensus building, and this standards and harmonization across global biomedical resources will truly, truly benefit from a partnership with utilizing criteria for better data sets and more solidified AI-ready data sets in biomedical research. I seem to be missing a slide with a plug, but I think it's probably because we did the presentation before I added it. My plug is join us at BOSC, Bioinformatics Open Source Conference, part of the International Systems and Molecular Biology Conference in the United States in Washington, D.C. So we will have conversations both about the role that artificial intelligence plays in the open science environment, and also we'll talk a little bit about sustainability and policies. Thank you so much for your attention. You can find our preprint with all of this information in that QR code, and these are our amazing collaborators. I just want to point out that we lost one of our amazing partners in all of this, and so with our work, we honor his memory. Thank you.
Thank you so much, Mujmani. Very impressive presentation. So we can see through the different presentation that AI will be one of the pillar of the biocuration. So be prepared, everybody. So now we are going to pass the mic to our next speaker. My colleague Henry will present the next speaker. Thank you.
Okay, the next speaker here is Mr. Wei-Keng Teo. He's going to speak about spatial transcriptomics, adopting a unified metadata standard and submission platform. Please join me and welcome Mr. Wei-Keng Teo.
Thank you so much, Henry. Tough first act to follow on Friday morning. Thanks so much. Great, really interesting talk. And as always, I think we're gonna bring the scale a little bit from like all of AI and bridge to AI to like one specific technique. So good morning, everyone. Hi, I'm Wei. I'm working as a senior bioinformatician at EMBO EBI, and I'm here to talk today about spatial transcriptomics, a technique that I think has developed and has really gained a lot of interest and traction. But I'll also give a brief introduction to spatial and then talk a little bit about the metadata standards. So for the brief introduction to spatial, in the 1990s, we start with microarrays. No, just kidding. I'm just going to quickly go through some techniques that I think give a little bit of context. So we really start with microarrays in the 1990s as a measurement of really measuring several thousand different gene expressions, but it's all bulk cell. So you have thousands, millions of different cells, and you're trying to understand gene expression. And this is really useful and really amazing. You can do differential gene expression and many other things. But it also has several limitations, which the next techniques will overcome. Then you go to bulk RNA sequencing, where you can sequence more of the full transcriptome, but you're still working on the bulk level. You're still working on the level of thousands or hundreds of thousands or even millions of cells. And one of the things that this affects you is that you really get this loss of information from averaging out multiple cells. This gray line here is sort of the average expression level for different genes, and you really miss out on different cell type populations, and you miss out on the precision of being and the granularity, because all you see is the gray line when you're working with thousands and millions of cells. You don't see distinct populations. After that, you move on to single-cell RNA sequencing in roughly around the 2009s. This is gene expression resolved at the single-cell level. So you're still working with, the transcriptome, RNA, getting a snapshot of what genes are being expressed and what proteins are being used in that cell at that time, but now you're at the single-cell level. So you're using fluidics and other techniques to capture an individual cell, and this opens you up to be able to find new cell types, resolve cellular heterogeneity, where you have this huge, complicated jumble of different cells and focus on one specific cell. Maybe you can find new cell types that were really rare and you couldn't pick out them in your bulk RNA sequencing. And then we move on to spatial transcriptomics. I really want to be careful here and say that spatial isn't necessarily always the next step forwards, because spatial sometimes doesn't have single cellular resolution. There are some spatial techniques that give you single cell resolution, but not all of them do. But what it does allow you to have is gene expression mapped to spatial tissue coordinates. What does this mean? I love metaphors. Bulk RNA sequencing. So if we take the example of kind of like your fruit tart or your galette, bulk RNA sequencing is taking this entire thing and blending it in a blender. You have the lysed cells, you have the DNA and the RNA, and in this metaphor, you might be able to pick out the vitamins and the fibers, but it's very difficult to reconstitute what originally was in there piece by piece, and you can't get any spatial information whatsoever. With single-cell RNA sequencing, you can pick out the individual cells. You can tell their characteristics, you can tell what's being expressed, so you know all of your individual pieces of fruit, but you're still missing as well that spatial context. With spatial transcriptomics, you have all of this information. You can put it together. You have the information on each of the individual cells, or at least a small cluster of 5 to 7 cells. And you also have the context of what they look like. We're three-dimensional beings. A lot of biology operates within this three-dimensional context, and it's important to be able to put this within that tissue context. This looks like, as a visualization, being able to take a tissue slide and being able to then look at your genes of interest on the left and say, cool, these are my genes of interest. Where are they expressed on this slide? Maybe there's a subtype here, a cell population I wasn't previously aware of. Maybe they're all grouped around the sort of emerging end of the intestinal villi. Maybe there's actually a method of me to track the gene regulatory network based on where different cells are expressing different parts of this sort of signaling chain. So this is my really pitch to you about how amazing spatial transcriptomics is, but also I want to talk about limitations, so that not to just jump on the hype train and give an unbiased view. Spatial transcriptomics can be expensive. There are new versions coming out which have more and more accessibility. I know that there are also groups here in South Africa which are working on spatial transcriptomics, and there's a whole number of different techniques. Kind of counterintuitively, the most expensive techniques, I would say, require the less adjustment. There are some machines that you can buy for, and they just sort of operate a little bit like a black box. But if you want to set up your own technique, of which there are many others, I have a list later on. I think it's really interesting, but you need to also invest in the skills and the knowledge to be able to adjust them to your own purposes. Cool. So Putting this into context of the data landscape of spatial, this is just a search about PubMed publications and literature in the last sort of like 10, 12 years. There's been an exponential growth of technology standards, published papers, conferences, and initiatives. AI is going all over the place. Spatial is also increasing, and a large part is this overlap of how do you make the data AI ready? How do you get this really gold standard, well annotated data that's really highly interoperable? And this brings us to a problem, I think, that happens in all sorts of different communities. And this is this problem of really, I call it heterogeneity, where you have diverse file formats. You have 5, 6 different file formats to capture the same amount of information. Within those file formats, let's say you pick out your file format, you say, I only want the HDF5 files because that's what I'm using. You open them up and they have different metadata field names. You have using different metadata standards. It's still not interoperable. You're working different sample donor tissue fields. They're all titled different things and it's difficult to harmonize them together. There's no universal data model or coding language. And so the packages that people are using can also differ from one to another. You might be a lab that's really well used in using Python, but not have that much R experience or vice versa. So it's really difficult to share this data around. And there's a lack of central deposition. You've got multiple different ecosystems, multiple different repositories. So people are submitting to different places, and none of this is really connected up very well. I work at the EBI, so we decided to tackle this from the aspect of deposition. So we are creating a home for spatial transcriptomics datasets, offering these archives and sequencing and imaging data. So this is a place where people will be able to come and submit and share their data. If there's one thing that the EBI has in abundance, it is different archives. So we have biosamples for metadata sample collection. This is a place where you can connect up all of your sample metadata, even and restrict your data if you want it to still remain private, but have a unique accession for your metadata samples. You can store your sequence data for spatial in the European Nucleotide Archive, which is a sequence data coordination platform. And then you also have your image data, which you can also store in the Bioimage Archive, which stores, indexes, makes all of this image data available. And our pitch basically is that it's free for anybody to deposit. It supports multiple linked data types. There's a funding strategy in place to support long-term storage because there's many projects have a beautiful website which showcases all of the data. And then at some point, the project funding runs out and that website still has to be maintained there. And maybe it's maintained for 5 years, 10 years, maybe another group takes it up to keep maintaining it, but maybe the website goes down. Maybe people move on to other projects and no one's looking at that email inbox. Whereas if you store it here, we can store it for free. And when you're thinking about imaging data sets that have like terabytes of data, storage costs start to really become more of a consideration. And our hope is that we can bring the community together around the metadata standard. But I also like to ground myself a little bit. I'm like, oh, wow, we could make a metadata standard. It would be great. We start with 14 competing standards and we say, we should make one that fits everyone's use cases, and then you get 15. So it's a difficult thing to do, and we're recognizing the limitations and the problems, and the way that we try to get around this is to engage the community. Interview scientists who are using this, ask them what they really need, ask them, hey, you're working with pathology, you're working in a hospital, you're working in Korea, you're working in South Africa, what fields would be relevant and useful for you, and adapt the standard to their needs. Collaborate with other organizations which have their own standards so that even if you're not one standard to rule them all, you can evolve in lockstep and evolve in collaboration. Collaborate with large consortia which have the data generators, which have the scientists, ask them to email out to their email mailing lists, engage with industry if possible, which is what we've done as well with 10x, to ask them to add some of these fields into their new output files so that when you push a button and you get an output file, that can automatically go into the standard. And then publish the standard and welcome feedback. So this is our portal. I've put the link up here just in case people want to have a look at it, but I also have a slide later on which just has a collection of links for people. This is where we are hoping to get access and submissions from different people, as well as showcasing our metadata standard, which is split up into different modules. Our metadata standard is something that we've been working on for a while, and we really hope to develop it more in collaboration together with more of the community, not just the people that we've spoken with, but also that's why we're here to get people's feedback if they're interested. Each sort of module has a number of different fields, a definition, some examples, whether it's imaging-based or it's sequencing-based. And yeah, thank you so much. And please contact us and give us any feedback if you have the time and also the interest. Thank you.
Thank you very much, Wei. He's actually from the EBI, UK. So the next speaker is Dr. Victoria Nongnes from the University of Padova, Italy. And he will be speaking on ordering the disorder. IDPO structures the knowledge on intrinsically disordered proteins. Please join me and welcome Dr. Victoria.
Thank you for that introduction. Yes. Okay, hi, everybody. You have me once again. Yeah, I'm going to be talking today not so much about the curation that I mainly work on, but on our latest updates on the intrinsically disordered protein ontologies. So first of all, I'm going to do a brief of a context, give you a bit of an introduction on what intrinsically disordered proteins are. Okay, so these are proteins that don't have a fixed 3D structure, okay? On the contrary of what you can see in a globular protein that has a defined shape. Intrinsic disorder presents themselves as highly mobile, highly flexible. They don't have, again, maybe 3D structure, so they may be lacking secondary structural elements or maybe tertiary. So basically, this intrinsic disorder can be presented globally and the full protein is disorder, or maybe it can be presented just locally and be present as an intrinsically disordered region or IDR. So as I was saying, these proteins are highly mobile and highly flexible. And because of these things, they're also very promiscuous interactors. They can have many, many different molecular partners, and they can bind to many, many partners at the same time even. So because they're so exposed to the media, being so flexible and moving around, they are also very liable to the consequences of these interactors. So they can also be subject to structural transitions. So maybe under certain conditions, these regions are disordered, but when they find a partner, they fall upon binding, becoming more ordered, for example. So on contrary of what was thought a long time ago, when the lack of structure was linked to a lack of functionality, we now know that they have a variety of functions, and they're very central to a variety of cellular processes and also linked to the development of different diseases like cancer, neurodegeneration, and even like viral infection. So there are ontologies that describe and capture protein functions, but not the structural plasticity that these proteins have, nor also the disorder-driven mechanism that makes these proteins very, very unique. The main home of IDPs is DISPROT, which is the major repository of intrinsically disordered proteins and regions. It's based on the manually curated data by professional and community curators, and it's based on the experimentally validated evidences about the structural features, but also the function features of these proteins. So not going to get too much into it, but this protein is organized on entries that relate to the different proteins that we have. This is an schematic, this is one of our entries, a schematic representation of the information that we have, and then we have all the different annotations. These annotations, again, are related to the 3 different aspects that we curate. So the structural states, the transitions that can happen, and also the functions. Disregard with which kind of annotation all the components of the information that we curate and add in this are the same. Basically, we go through a publication and we collect the reference. We analyze the experimental approach that was done and what this was evidence for. What is this telling us about these proteins? And we capture this with different control vocabularies. So again, we have a variety of control vocabularies to express the things that we want to say about this protein. So we could be talking about the structural state. Is this disorder? Isn't this disorder? The transitions that can happen between disorder state and order state, for example, and also the functions. The functions, we have two different groups. We have one group that is like disorder functions, are very much related to the disorder nature of the protein region that we are describing. So things like, for example, entropic chain, or even like self-regulatory activities that are very related to our scope. But then we have other functions that we can annotate in this plot, like more general, more common, like the gene ontology functions. So of course, for the gene ontology functions, we relate to gene ontology terms, but for the top part of the descriptors, we use IDPO, which is the intrinsically disordered proteins ontology. But to be completely honest, until today, I mean, today we're still doing this, it's not a complete organized ontology, it's more a control vocabulary. So we set up to update this and make it a formal ontology. And why we did that? Because ontologies make FAIR possible. We want to have a database that is fair and data that is fair. So we wanted to start by making our ontologies fair. So we wanted to provide and standardized vocabularies and annotations that make data findable. We also see that ontologies make FAIR possible by defining clear relationship and meanings, ensuring that the data is understandable and, because of that, reusable, and by enabling integration across different databases and tools so the information can be accessible and interoperable. This is the storytelling in a way. Actually, what happened is I wanted more terms. And we were like, okay, let's add new terms. Okay, but shouldn't we reorganize this? Okay, but shouldn't we? And they become a whole thing, snowballed. And we decided to be, okay, let's aim high. Let's try to formalize this. Let's submit it to our OVO. Let's apply also the OVO foundry principles and make this a formalized ontology. So basically, we are doing we've done the updates. On the first thing we managed or we were trying to do was to add these terms that I wanted related to condensate and phase separations. These are phenomena that are very, very much related to intrinsically disordered proteins, and we weren't able to capture them the best that we could. So we decided to add a higher order assembly transition and some terms that are related to biomolecular condensates, liquid-liquid phase separations, gelation, crystallization, and so on. And we also reorganized one of our disorder function branch that was the self-regulatory branch. Now we reorganize it in a way that it has a different parent. Now it's self-interaction, a more broad term that can not only contain self-regulatory activities, but self-assembly. And we also did some housekeeping updates, let's say. So we redefine every single term and provide synonyms for them. We moved to a 7-digit identifier format. We used to have 5 digits, so we had to move for 7 because that's the OVO Foundry community standard, so to prevent ID collisions across the different ontologies. We added lookup axioms, so to enable automated reasoning and inference. So now disorder to order, the transition has as an initial state, the state disorder, and as a final state order. This might seem trivial, but it's really important for the organization of the ontology. And then we also add integrated to BFO, to basic formal ontology. So now our major branches map to quality, process, and function terms from BFO, which enables cross-ontology compatibility. So because of all of these updates, we moved from IDPO to IDPO. We have the new terms here. This is a way simplified version, of course. But just because we have new terms here, we have the restructure, and we have submitted to Abo Foundry, and we are on the feedback loop. So they're getting us some comments. We are trying to apply changes or explain better our reasonings. Also, IDPO is now openly maintained in GitHub, where anyone can access the releases, propose new terms, report issues, or just join the discussion. We have it also available in a variety of formats, so to also provide interoperability. But of course, even if it's not yet approved, we are still thinking ahead on what we want to do on the future for IDPO. And we want to expand beyond what Disprot needs, because of course I'm Disprot, so I'm thinking about Disprot, but there are really a variety of different IDP resources. And we would like to capture them all, so to actually have a cross-database interoperability between all of these resources. Also, we need a new, more mechanistic terms. For us, this is a huge thing because, as I was saying before, the context plays a major role in intrinsically disordered proteins. So we need to capture these kinds of folding up and binding or ensemble descriptions. That for us is very, very important. Of course, we need to update the curation guidelines and add how-to documentation for our curators, because this and many of our resources are community curated as well. So we need our curators to know the updates and how we are going to be adding these terms and these definitions. And of course, eventually, do a formal publication of the updated IDPO, and in which we will be able to assess also the impact on the IDP data and standardization. So this is all for me. I would like to thank you all for your attention. I would like to thank also my colleagues from the Biocomputing Lab at the University of Padua, especially Ximena Castronese is the PhD student that took on this and dived fully. We are not actually we are still learning, know much about ontology development, and she just went for it. She created the repo, she did a bunch of things. So this is a lot of her work. And also I would like to thank our collaborators across the multiple disciplines and different parts of the world that help us also with different challenges, each of them. So thank you again.
Yeah, thank you very much, Dr. Victoria, for that work on IDPO. The next speaker is also from the University of Padova, Italy. And he'll be speaking on prediction of intrinsic disorder function in the critical assessment of protein function annotation. Is Professor Damiano Piovesa. Please join me and welcome Professor Damiano.
Okay, good morning, everyone. Thank you for being here. So today I'll talk about predicting intrinsic disorder function in kappa. And also I'll be talking about standardizing evaluation and what we learned from IDPO. So it's very connected from what you saw before. So this is, of course, working collaborations. Here you see the list of people who contribute my work. So intrinsic disorder proteins plays a lot of roles. I mean, you've seen the presentation from Victoria just a second ago. And these functions are captured by the intrinsic disorder protein ontology. So on the other hand, we have kappa, which is a community challenge to evaluate protein function prediction method, which is generally based on gene ontology annotation. So essentially the task is, given a sequence, tell me what is the subgraph of the gene ontology that's associated to that function. So the question is, can we apply the kappa environment, kappa infrastructure to IDPO? So the problem is that kappa evaluations are hard to reproduce and compare. So for instance, here you see the results for kappa 3 editions. So you see precision record parameters. And at the time I was participating as a submitters, and I had in hand my predictions. I saw the plot in the paper, and I tried to reproduce the curve. Actually, it took me a year to test all the possibility, to test all the choices. So for instance, understand if the root terms are included, if cross-ontology relationships should be considered, how to manage alternative and obsolete IDs, how to propagate the scores, how to calculate information content, how to weight terms. So this is really hard. And for example, if you think about something simple like normalization, so for instance, if you want to normalize just considering predict proteins, or if you want to normalize just all the proteins that you have in the ground truth, this is what you will get. So 3 different curves with the NAF score, which is the weight, the average between precision and the call, that's differ significantly, even for a super simple case like this one, where you just have a bunch of nodes. So really, these are things, a lot of choice can affect significantly what you see on a plot and what you will evaluate and how you will evaluate function prediction method. So my contribution, the way I tackled the problem, was by developing a new piece of software, which is called CAFA Evaluator, with the aim of standardizing the evaluation procedure to make the results reproducible and to let the user to choose among different options and the things that you can modify for getting different types of precision of the core curves. And of course, it's super fast. So it's quite popular. It has been forked in the star many times in GitHub, and now it is part of the official CAFA challenge. It's been adopted in the CAFA infrastructure. So CAFA now is run on Kaggle, and if you submit your prediction in Kaggle, there you get immediately placed on the dashboard and ranked among the other methods. So it enabled a consistent benchmarking, and as a side note, it led me to become a co-organizer of CAFA. So just a bit of context. So CAFA has been running for more than a decade now. So the last publication dates back to 2019, so corresponding to CAFA 3. And since CAFA 4, it has been done in-house. So the organizers actually provided the evaluation by themselves. And since CAFA 5, the challenge was moved to Kaggle. And CAFA 6 is ongoing. So you will probably see the results at the ISP conference in July. So the Intrinsic Disorder Challenge was launched in CAFA 4, so in 2019. So this is just the submission deadline. And since then, I mean, we redone the evaluation, and we've been able to collect data for 6 years of annotations from Dishcrot. So data collected from crowdsourcing, so Victoria mainly also. And we evaluated 3 different branches of the ontology. So disorder functions, binding pattern, and structural transition. Binding pattern actually now it's fully mapped one-to-one to the gene ontology, but at the time it was an aspect of the IDPO ontology. So we evaluate predictions that predicted terms as if they were in the IDPO ontology. So with this body of annotation, which is huge, we managed to provide a reliable and significant evaluation. So the results. This is the surprising thing. So if you look at the results, the striking feature of these curves is that the best method is the naive predictor. So naive is just a method that transfer all the terms to all the proteins in the ground truth with the same probabilities, and those probabilities correspond to the frequencies that you see terms in the training data set or in the old digital database. So why is that happening? This is not something new. This was observed also before in the regular function prediction in the regular CAFA challenge. especially with the protein binding term. So protein binding is very frequent, and it affected the performance of naive. So in CAFA, they solved the problem either removing protein binding from the ground truth or weighting terms based on the information content that they have in the database or in the comparing it with the ontology structure. So we analyzed that, and actually we have similar problems. So the number of terms is not that high, so the ontology is small. You see the frequency of terms on the right, and you see that the distribution of terms is dominated by just a few of them. For instance, linker or regulation of phosphorylation and so on. So you see here also the accumulation of annotation after you expand terms annotation up to the root of the ontology. But on the other hand, what you can also note is the information and how the information is distributed inside the ontology. For instance, you can notice that there are the most informative terms. So this represented the information gain or the information accretion. So how much more information is provided by a term compared to the information that's provided by its parents. And you see that the most informative terms here are already there, but it's not the only problem. The other problem is that the information is concentrated on terms that are close to the root. So in the gene ontology, this is different. Most of the proteins are annotated with leaf terms. And if you calculate something like that for the gene ontology, you will see that the most informative annotations, not all of them, but are distributed towards the right of the graph. So the key insight here is to compare CAFA with another challenge, which is called KIDE, which is critical assessment of intrinsic disorder. And in KIDE, we evaluate one function at a time. So essentially, we evaluate prediction methods that predict, for instance, if a region is binding something or if a region is a linker. And we consider positions in the sequence. So we evaluate where the region is predicted, and we evaluate, we calculate the confusion matrix at the residue level. On the other hand, in CAFA, we evaluate multiple functions at the same time, and we ignore positional information. Gene ontology, by definition, is gene product ontology. Is not based, it doesn't describe the function at the residue level. So this was sort of expected. But for disorder, since disorder is region-based, this difference is critical. So to conclude, there are 4 take-home messages. First, standardization improves reproducibility. Second, CAF evaluator enable consistent evaluation. Third, not all ontologies fit CAF assumptions, and you have to be careful with that. And the IDPO at the moment, given this topology and given the distribution of the notation in the available database, requires alternative evaluation strategies. So with that, I'd like to thank all my collaborators and thank you for your attention.
So thank you so much for all the speakers. So please, can you join us for the panel discussion? So great talk, everyone. I really enjoy the diversity that we have in this conference. So thank you, gracias, grazie mille. So we will open the floor for question, but as a chair, I'll be the privilege to start. So yeah, Wei, I think what you did present it's science fiction for me, but hopefully this kind of images with expression will be some routine thing. So I think it's very emotion field. So my question is for yeah, Victoria, thank you for reminding me how building ontology is quite painful, and having all this discussion with Obo Fundry. So every time you have a lot of mailing of critics and requirements. So thank you so much. So my question is for Moni. So do you have any training or capacity building for the AI readiness implementation? So are you planning to do such kind of training? And the second questions, are your tools are adapted to, let's say, for example, the African context, and if they could be adapted to the African context? Thank you.
Thank you for that question. Wow, that was loud. I'm sorry. Um, Also, thank you to the sound booth because we have return here and I can hear everybody much better today. Thanks. Thank you for that. Your first question was regarding training and capacity. So at the moment we have, it's all self-evaluation and instructions available on GitHub, but we are planning to have some workshops that allow us to start training the community with this. At the moment, you can just reach out to us and we're happy to, the spreadsheet that we utilize for self-evaluation is available in Zenodo.
You have the community over here for Biocuration, so I'll be very interested to follow the workshop.
Exactly. We could, I see that there are now some webinars that Biocuration Society has been doing on a regular basis. So we could organize one and anyone can join online and we can just go over these principles. So I love it. She's already taking notes. Let's do that.
Yeah,
Yes.
you have the society.
We'll
So
be accountable to that. And then that'll be a great opportunity to capture some feedback from you as well. And then your second question was on whether this is generalizable to the context of research in
African context, for example.
There is no reason why it shouldn't be. So when we think about these characteristics, we're specifically characterizing the data, where it might be, where it will definitely need some translation to the context will be when we start talking about the ethical principles. So you can see, so we have made our best attempt to making sure that we observe guidelines such as the regulatory and ethics workstream guidelines from the Global Alliance for Genomics and Health, which takes into account a global context. But there might be some regional things and regional expectations about how do you adhere to ethical principles that may vary. And so it would be really good to explore that. I have not done that exercise. And I think if this is something that is necessary, then we should probably figure out how to update and expand on the criteria to adapt to that context.
Great. Thank you so much. So any question? Yeah. So for Alex.
Thank you so much to all of you for your insightful presentations. So my question is for Monica. From a data curation perspective, how do you balance data standardization across multiple resources with IRB constraints and data accessibility?
Thank you. That is such a wonderful, wonderful question. Thank you for asking this. So we have actually been working very closely in trying to figure out, follow the ever moving target that is the data sharing and dissemination policies of the United States National Institutes of Health. What, to the best of our ability, we make sure that we adhere to the latest in that aspect. But at the ground level, right? Like, how do we actually make sure we do our best to just leverage current practices across? And we have consulted with experts on ethical, legal, and social inputs. And we have actually a working group within the Bridge to AI that focuses solely on the legal issues across the United States and Canada. This project is funded in the United States, and it's focused. The data generation is focused only on institutions located within the United States and Canada, but the practices are as broad as we can make them and as generalizable as possible. So we just keep an eye on listening what are the latest broadly accepted practices. We keep an eye on what, we're in the US, so we're HIPAA compliant type of ethical principles and guidance, but we also pay attention to what GDPR is sharing and how other institutions are working. And then we also extend partnerships so that we make sure that we're talking with the same language. So for instance, I have been already in contact, and it's been really satisfying to see that both the UK, the AI Bio UK group utilized our criteria to build their own core. We also have been talking with the Elixir folks, the AI group in Elixir, to making sure that we are talking the same language when we're trying to specify these guidelines and there's criteria for AI readiness.
Thank you. So, Alex?
So
then
Damiano, thanks for a great talk. I was interested in the assessment of this. So for the globular proteins, UniProt sort of curates Go terms, and then those are used, they're sort of held back and used. So can you explain how this is done for the disordered portion?
You mean the annotations that are available in UniProt?
No, how the terms that were assigned for the testing, is that sort of disprocuration, and do you hold that back? How does this all work?
Yeah, we got all terms from Disprot, and the weird thing is that challenge was done on 2019, so people submitted prediction in 2020, and then we evaluated the predictions after 5 years, just because we keep writing the paper for 4 years, and over the years I recalculated the evaluation as soon as new annotations were accumulated. So that's why we accumulated that many annotations. But the idea was to rerun the challenge after a year or so. So that's the only reason why we waited 5 years. I mean, both papers, CAFA 4 and 5, are ready. We're just, I don't know, waiting to submit them.
Nikki, then Barbara.
Yeah, thank you. Hi, Nikki Tiffin from Sandby here in Cape Town. So also for Monica, sorry, you're being picked on a bit. So I wanted to ask you about your AI readiness criteria. I didn't see equity in there anywhere. So you have ethics, which is kind of about participant protection and sort of the do no harm stuff. And you've got provenance, which is about looking at what we did. But I didn't see anything reflecting beneficence and checking what you're giving back and checking who's benefiting and checking how they're benefiting and is it fair and equal, which is different to bias, right? It's a different thing. So yeah, I would be interested where you see equity. And I guess following on from that, I would ask you where is your Global South representation in your stakeholder engagement in your AI readiness criteria? Thanks.
Thank you very much for that question. First of all, I want to make sure this is a disclaimer. I do not speak on behalf of the United States National Institutes of Health, and I do not speak on behalf of my institution. I speak on behalf of myself. In the current climate, that's necessary. We actually have these aspects of equity. If we look at this, the spreadsheet very closely, you can see that we have made considerations for the protections of persons. What is the significance of the type of what kinds of principles have we observed, whether there is information or not on where did the data come from, who are the persons that the data came from, what kind of consent was captured, what kind of access people will have to the data. So all of these questions we ask, and then we can display all of these on the data sheets. I'd love to sit down and share with you those more detailed rows. When we talk about ethical principles here, I only speak about provenance and just making sure that the data was ethically acquired as a generalization, but there's a lot of depth to that as well, and we have made considerations for this. And then where is the Global South? Well, I traveled to South Africa to open this to the connection to the Biocuration Society and to express, this criteria that we're just starting to socialize across the globe. It started as a project specifically funded in the United States, but we believe that the criteria are generalizable to all projects that deal with biomedical data with the good advice from our colleagues on where we could be expanding them. I'm making myself a to-do note regarding some webinar explanations, and I think that may be a good way to share it. And then the other thing that we haven't started, but I think would be very useful, is to start thinking about the potential for translating the criteria into other languages so that we can provide it into other more regional ways. I'd love to welcome your feedback and give me a hand on how we could do this better. Cheers.
So, sorry, Yasmina, just Barbara is still waiting for the beginning, then Yasmina. Go ahead, Barbara.
Okay, so I also have a question for Monica and also about AI readiness. So we are all talking about making these AI ready data sets for machine learning, and we need metadata. So are you aware of initiatives that define what is the minimum level of metadata we need? So I think we need to work together with machine learning scientists as well. I know this has been discussed for so many decades in very similar contexts, right? Was this MIABE initiatives, the minimum information about the bioactive entity. I think this it will now change a bit because the way we use the data is a bit different. So are you aware of initiatives and can you comment on this?
Us. 82 top-level classes to describe metadata for clinical data sets to make them AI ready. Absolutely. So I brushed over this, but in one of the slides I was sharing that we've developed some tools for both the capture and the dissemination of metadata about the data sets to be able to grab all of this information. So we have worked with the data generation projects in our group, but also with other domain experts to create a minimum metadata schema. And we have generated this both in arrow crates and LinkedIn serialized it into LinkedIn for a semantic layer. And then we have also incorporated principles from Croissant for the RAI, the version of the metadata schema from Croissant, and have been conversing with, like I said, Elixir Europe and UK AI Bio in order to make sure that we are taking into consideration what the community has been using for standardizing metadata in the clinical space. And so reach out to me. It's on the slides when you see a little bit about Fairscape and generating our crates. But definitely, I want to make sure that we more prominently disseminate that this is an effort that we're starting or that we have been carrying for a few years and want to make sure that is more globally accessible.
Thank you.
Cheers.
Hi, thank you, Malik. My question is to Dr. Victoria and Professor Damiano. Thank you for the very interesting subject of disordered proteins. I'm just wondering, I don't know very much about them. Roughly how many are there? And is it like they are sometimes disordered, sometimes stable, or are they always disordered in the all environment, cellular environment? And are there any motifs, some kind of sequence, structural motifs that they have?
Thank you for the question. Oh, that was loud. Sorry. We don't know how many there are. I mean, in this plot, we have information for about 3,000 proteins. They're probably not. They're definitely way more. But since their physicochemical characteristics, they're very difficult to express and purify. They also get proteolized or get aggregated. So some of them are very difficult to assess and to properly say, okay, this is disorder. Of course, now we have AlphaFold that predicts, I don't know really, maybe Damiana knows, how many of the whole proteins are predicted to have any kind of disorder. AlphaFold is not so good on the disorder predictor, but can predict when it's not so sure about the structure, so I don't know. Regarding the other point, there are proteins that are fully disordered, and they stay disordered. At least we don't have evidence that they gain a structure, but there are a variety of other proteins that have regions that are disordered and change and get order, or even the other way around. They are fully structured, and because they are changes in the pH that are relevant to the work of this protein or whatever, they lose the secondary or tertiary elements and get disorder as well. There are some motifs. I mean, the disorder has some relation with the amino acid sequence, of course. There are no motifs, but there you can see like a bias on hydrophilic residues, for example. So you can spot or kind of think that there could be the disorder. And we also have, regarding motifs, there are some motifs on disorder regions that are very, very well studied, let's say, or described for binding or for modification. So we have like our own IDP sphere of motifs and tools and concepts, let's say. I don't know if okay.
Thank you, gentlemen.
Thank you. Mine also goes to Dr. Victoria and Professor Damiano and disordered proteins. I'm wondering whether there is a possibility of a disordered protein changing to disordered, to ordered states, and how would the ontology or the database address that?
Yeah, this is captured by the transition terms. So transition terms should tell you if a region transits from order to disorder or the other way around. There are also intermediate states like molten globule. This is something in between. I can also answer to the previous question about the distribution of disordered proteins at the database level. We also maintain the MobiDB database, which is based on predictions. According to predictors, AlphaFold is not the best to predict disorder. It's on the top 10, but it's not the best, according to Kite. And it's suspected that 30% of the proteins on higher organisms like human have intrinsic disorder regions. So it's a very common thing, and it's connected to regulatory processes because they are extended, they are very accessible to enzymes and interactions with other proteins. They are highly modified, so they're really important and they are understudied both from the functional point of view and also from the structural point of view because we don't know actually the dynamics. All the experimental evidence is indirect because we see, for instance, missing residues means we don't see these regions. We can measure what is the average distance between two residues and these other regions, but this is an average measure, like coming from NMR and other types of experiments. So it's really an open field, and I'm very interested to see what comes in the next years.
Any other comments or questions? So we have 2 minutes left. So a last comment, I would like to highlight something that is quite interesting through the different presentation for each of you. So we noticed that everyone is working on metadata standardization or ontology development, as you mentioned. Jimena is the lady who's working on it. So everyone have no background or knowledge before about data curation and ontology development, as in my case too. So regarding this, I think the society could do better to create a new community and maybe offer some kind of training as well, capacity building for data curation, biodata curation. Let's say ontology tools is one of the common tool for harmonization and data standardization. So I think that even we starting to do this without no knowledge before about data curation or basic skills about data curation and ontology development, that could be quite interesting. To think about it. And this kind of conference is a good occasion to create communities and networking about. So you have contacts about people, you can just create new training materials for people interested in biocuration. I think, and everyone working with data and biodata should have this minimum that we were working with minimum data. I think we should have minimum skills to work with data curation. So any comment or so thank you so much. We can close the session. So we have
Thanks for having us.
thank you so much. Thank you so much, everyone.
Welcome back, everyone, to the plenary session. Could I ask you all to settle down? Okay. Oh. So welcome to Chiamo Wasegwa. So today's plenary on the African Open Science Platform, implementing African Open Science Vision. So Chiamo will have 30 minutes for his presentation, and we'll have 15 minutes to pick his brain. So let me let me introduce Chiamo. Mr. or Dr. Mutsewa is the director of the African Open Science Platform, with a strategic portfolio to direct and support the platform in its mandate to advance and coordinate and amplify the impact of open science in Africa. So Chiamo received his PhD in computer science from City University of London, UK. And of course, he has multiple responsibilities in this very... Too long. Just, you see, he wants more time for himself to talk, so I'm going to leave it to that. Yeah, so you want to take the time. Okay, thank you and welcome, Chiamo.
Thank you. Everybody sit down.
The sticker?
This is the one. Okay, thank you very much. I'm so excited to be here. I don't even think I need slides. And I don't think you will be picking my brains. I will be picking yours. I have a lot of slides, but it's not so much about the slides, colleagues. I attend so many of these conferences. We talk, we have slides. What is important is the dialogue that we have to have together and the strategic directions that we map. I think I have the best job in the world. I'm a science diplomat, the one who ran away from doing science, because the environment that you do science in has to be fit for purpose. And I thought it would be very important to, I wouldn't say sacrifice, but I would say to complement my career, to be able to engage in other fora, to make sure that we can do science, obviously that is rewarding, impactful, but also very relevant. So when Nikki and her team approached us last year, I think as far back as August or September, just to pin us down to a keynote, I could not resist. Colleagues, the AOSP covers all those grounds. I don't host your data, but I'm an open science diplomacy platform to advance all those areas that you see there. And colleagues, you know, sometimes we thank people after 40 or 50 years of work. For one reason or the other, I've always gravitated towards biologists. And I want to highlight three people. Obviously, at the very top is Nikki, whom we are very appreciative to have in the continent. Do you remember the History Africa project, the History A Bionet, the ILOAZI, and now the ABI? These are huge projects of strategic importance to Africa. So by no small measure due to her efforts and her community and her colleagues. So let's take that time to really put her at the top. A distant second is Barant, that you all know very well. There was a talk yesterday about malaria. Barant, I always tell him that he's an African at heart. I think he spent a lot of time in Kenya. And as you know very well, he's the pioneer and one of the co-authors of the FAIR Principles. And he was the president of the ISC Core Data. And we were together in Leiden at the Lorenz workshop, reflecting on the 10 years after FAIR and what should happen next. He's pretending to be retired, but he's still doing a lot of work that I'll show you shortly. And I think this is great to have scientists who can connect the dots, because colleagues, we're increasingly asked to show the relevance of what we do. I appreciated the question that was asked by a student who was here on day one after hearing all very complicated talks and asked whether we are doing any medicines or therapeutics from what we're doing. A very distant third, allow me to take this. I mean, if I run out of time, it's fine. A very distant third is a guy who happened to be my supervisor. He came to City University in 1998 after his postdoc. I think he was in Aachen, and then he went to British Telecom's labs, then he joined City. He's a computer scientist, a computational logic. First year
of my PhD in 2001. He said, Chairman, you can go to British Telecoms. He arranged for me to go there. And when I came back the following year, he took a sabbatical. He went to the US. This is the early days of bioinformatics. And when he came back, he says, guys, he had 7 of us as PhD students. I got good news and bad news. Good news is that all of you have got very good topics. Bad news is that I'm leaving. I'm going to Germany to take a professorship at TU Dresden. Before then, he tried to convince me to get into bioinformatics. I said, no, this sounds like an imposter syndrome where computer scientists want to become biologists. And I was very sensitive. Colleagues, he's done fantastic work since. I had some of you talking about PubMed yesterday, protein-protein interactions. And here was a computer scientist with a computational logic developing tools to assist you do the work. I'm saying this because there are some young scientists here. I can see some of them. I was speaking to Umesh yesterday. You can build the networks that Professor Lovering was talking about. These are some of the networks you need to share with you. And I think now he runs a very large group and some spin-outs in Germany. So there are some of the connections. Colleagues, the biological sciences, medical sciences, together with the astronomical science, you are doing great work in biocuration. And rightly so, because this is it. You had the presentation by Blessing yesterday about the data gaps that we have, especially with strong implications to the accuracy and the representation of the models that we have. A lot of work has to be done. And we are doing great work, turning obviously data to data assets that we can then use. And rewarding people for doing the work that they are doing. Why should somebody invest a bulk of their time doing data curation if in the end we just sort of recognize the publication alone? Publication is critical. Hence, I think things like the credit taxonomy allows us to purposefully mention all these roles and people who do them in the work that we do, so that at least there's a record that you did this work. Of course, this talks to the bigger picture of why it's time to reform research assessments accordingly, infusing it, reforming it, and embellishing it, and improving it to obviously skew us into the right thinking. Of course, there are things that we need to do to be able to, and it takes courage. A big ship has been sailing for so long, you just want to turn it a little bit. What are those key things that we need to do, if at all we agree that this is the way to go? I just put those things there to stimulate your thinking. If I run out of my talk, time is fine. I'll give you the slides. I wanted to pick your brains a little bit, especially the young people. I know that you cannot teach an old dog new tricks, but it's not too late. The data steward, everybody talk about AI readiness of data. There's no AI without good data, good quality data. And surely it's a no-brainer that the data steward is at the core. And how do we train data steward with the right competencies, considering all those expertise areas? And how do you professionalize your profession and have a career path in these areas? So I'm highlighting a few things on the right-hand side there. Again, Barand and others have been engaged a lot in this work on data stewards, Fair Connect, and I'm sure you heard about nano publications, that you can do these small publications, have your research findings, as small as they are, represented as machine understandable nano publication. You can do a nano publications out of your presentations that you did in this conference. So very important work that will also allow you to have a digital trail of the work that you do. I live on a suitcase. So I'm very happy that Nikki and the team brought this conference to Africa. I always stick out as the sole African in Europe in some conference. But you can see I'm not intimidated. I go there and say the same thing. These conferences must come here for you to enjoy what you enjoyed yesterday. They go old, and you know the wine is not bad. But I really appreciate all the international colleagues from all over the world who came here today. Conferences are a very good fora to also build relationships between people. There's very good work. I'm just putting this to also connect the dots with your community. The work around fair digital objects. We talked about the internet, all these linked web servers with HTML pages that you can get to using protocols like HTTP with TCP IP underneath. How can we elevate the conversation now to have this web of fair digital objects and create these data spaces? And of course, if we do, there's opportunities to address these principles that are now being floated. I don't think they're published yet, but Peter Wittenberg and the others are pushing for them. And the very guys who actually invented the internet, if you want to call it that way, they are part of the Fair Digital Object Forum, and they're pushing for these principles. I was invited to Vienna to come and provide this African inputs. I said, I can't provide African inputs. I can provide my inputs and airspeed until I engage and solicit inputs properly. And this is exciting because you can build these trust frameworks, embedding the STAB principles into FDOs. And you can see the last sentence at the bottom there. We can integrate these STAB principles into FDOs and really do a lot regarding developing these global ecosystems you're talking about. I will also encourage you to read this particular publication. It's the STI Outlook. Happens, I think, every year. South Africa hosted a workshop to launch this for South Africa in terms of the survey that was done. This issue of secularization of science, critical. This issue of making sure that we get the balance right. Trust is there, reputation there, dual use of technology, science diplomacy is very critical. So I think as and when you discuss, I know that some of you yesterday said they're only dealing with plants. They're not worried about data sovereignty and others, but it depends what type of plants you're dealing with, isn't it? Especially now there's also conversations about intellectual property regarding some of the plants. Colleagues, I'm saying this because we are talking to the converted, but when you go to policymakers, they push back, like it or not, on data sovereignty. And you can see where they're coming from where they're sitting. So let's not pay lip service in our communities when we deal with data sovereignty. Bottom line is we also need not just convince ourselves, but those that are also funding research, all this fits together. People are talking about data visitation. I remember the early days when I was doing my PhD in multi-agent systems, there was talk about mobile agents that can propagate around the internet and sort of visit data. These are not new ideas, but they need to be implemented. Data visitation. I mentioned the work that Barant is doing in his retirement. He's got this slide. I just emailed him to say, can I use it? He had a presentation at our African Open Science Governing Council. They're pushing, when I say them, the community is pushing for this data visitation as the way to go. Let the data sit where it sits. Let's provide a mechanism to access it where it is at. Surely with data sovereignty, there are layers, policy, technology, and other arrangements to address it. It's an avenue. It will allay maybe some fears, and it's something that is being discussed. And of course, they want to create an ecosystem, including with industry. You know, there's always this natural tension between academia and industry, but there's some efforts to do this. And the last time we spoke, even when they were launching this, he said, look, we need Africa in there. I said, look, I'm not Africa. Talk to the domain expert, talk to the community. So that's why he put that badge there. I don't put our badge there. Just go talk to the community. This is a strategic area for Africa. It will address that challenge of data sovereignty, if there's a technology solution. And obviously, metadata has been talked about in these conversations. You guys are experts. You can see what is required. So I'll point you in this direction and the community working on this for you, maybe if you're not already engaging, to explore avenues for doing it. I have to put this because during the South African presidency, in as much as there was a lot to discuss, the South African presidency wanted to highlight some areas of strategic importance and scope out some deliverables that during the presidency, they could be advanced. Obviously, we heard about the theme for solidarity, equity, and all those. even though there wasn't a concrete deliverable eventually, the dynamics of these multilateral platforms in terms of agreeing on deliverables can be protracted, but at least there was a thinking around these consultations with stakeholders like yourselves around avenues, including with capacity building, and really potentially unlocking the value that we all agree is in the genetic diversity of the continent, and that the continent also has to play a major role in these endeavors amidst the challenge that we have. I really want to highlight this to you. I'm sure you already, in your own way, in your own community, abreast. Colleagues, data is at the core, and I reiterate this because you have a data conference here. There's a huge conference that is called the International Data Week. The immediate previous iteration was in Australia. As ASP, we engage in these platforms to make sure that the conversations around relevant issues for Africa don't go through the cracks. So we hosted a session there on infrastructure, the possibilities of developing this policy-aware federated infrastructures collaborating with everybody else. I mention this because there's something about Cape Town. I don't know whether it's the wine, it's the food. It's coming here, it's coming here. So put it in your calendars. 2027 look far, but it's not. When I say I would like, because I'm in the committee, I would like to see this community here having a very robust pre-conference events. You can have these pre-conference events. You can have sessions at SciDataCon, and you can certainly have papers. It's the biggest data conference in the world. You can see they have highlighted at the bottom left. The theme, in a way, even though it's still evolving, is open data, open science for global solutions. I mention this because the value has to start coming out. Some people are saying we talk too much. The policymakers want action. We must demonstrate value. Colleagues, I mention this because those who are old enough in 2018, just the second conference in this iteration, right after the one that was in the US in 2016. I forgot where it was. It was in Denver, I think, Colorado. The second one came to Botswana, and I was in the committee. And the first thing we recognized then was because we were struggling convincing the policymakers to really put this conversation around policy frameworks that can support advancement of open science. We fought very hard. I recall that we fought very hard to try to get to the higher office. Eventually we did, because prior to that in 2016, we hosted the International Conference on Cybersecurity, and we helped Botswana develop the cybersecurity strategy. So eventually you can see there, no list at the very front, we have the president, some ministers, some DDG with a beaming smile, who was actually, I'm saying this because he's no longer a DDG. He made it very difficult for us to get funding. But I think with the last week to go, he got funding. And I have to say, colleagues, engaging these policymakers opens a lot of doors because they own the conversation and they can put it in their priorities. I'm not going to say what happened. What happened in Botswana stays in Botswana. Fantastic. We talk about indigenous knowledge, equity, this equity. The care principles were coined in the margins of the International Data Week because you made it purposeful to bring this to the fore, not just for romantic purposes, but putting the case forward for things like the bioeconomy, which I think you should also think about going forward. So the care principles, I think a lot has to be done in implementing them, especially in the context of developing countries, because there's knowledge there that needs to be brought to the mainstream of science. From here, with my science diplomacy hat, I'm going to Addis Ababa at the AU, UNECA, UN annual calendar events, colleagues. This is the forum where everybody goes, including the policymakers. The ministers sit there. Last year it was in Uganda. The Minister of Science herself is an epidemiologist, and she hosted that fantastically, bringing the conversations around science, the science policy interface, and societal impact. I'm saying this because I must come out of here. You're not going to pick my brains, but you're going to tell me what to go tell them, so that you're not an obscure
little conference in the corner somewhere in Cape Town or wherever it is going next. But we are bringing to the fore what those stakeholders, including policymakers, need to think about. I generated these images, and I'm thinking to myself, how nice would it be if there was a policy brief, very small 2-pager, that you can give to a minister and say, these are the opportunities through biocuration for the bioeconomy in Africa. I'm not saying you're in the business of job creation, but you're in the business of situating what science is and what it can do. And you can also obviously talk about the opportunities for biocuration in Africa and the partnerships we need because we are lacking. That's an example, colleagues. I don't know, have I run out of time? How much time have I got? 10 minutes, okay. So really that's what I was gonna talk about next. But I thought the previous part was quite important, just to ground us a little bit. I know that we are focused on our science and our next promotion up the ladder depends on what you produce, but there's a bigger picture here. I'm sure that you know very well that funding is becoming constrained. NIH funding is drawing down, climate change funding. These are decisions above you, but you must constantly fight your corner. So when it comes to open science, the good news is that the countries agreed to this. You'll not be making anything up new. They agreed this. In fact, they are going to report progress every 4 years, which is good because as part of the system, there's need for reporting. Again, these are slides I got from my UNESCO colleagues. What I like is, especially for the younger career researchers to appreciate this, there are all those values and principles. Obviously, there's an interplay between them. And as scientists, you know very well, deep down, you know very well, and professionally, you know very well, the science that you must produce must be reproducible. And that is the underpinning of integrity. Yeah, so the principles of transparency and scrutiny through peer review and reproducibility and critique by your colleagues manifests itself through open science principles, through the conduit of open science too, high quality research and integrity. Of course, you can do the same exercise for all those. And you also see there the need embedded in the definition of open science for including other actors in science. Science is not just the purview of scientists. And you know this very well in health. You are doing epidemiology, you are doing vaccines, you are doing pandemics, HIV. It's not just your science. You talk to the community. You should have communications mechanisms to translate your results. Science communication. It's not just you in the lab. You saw what happened with COVID, that interplay. So you can see that we need to think deeper. Obviously, a quantum physicist may argue strongly how they will do it, but I'm sure if they use their big brains, they can figure that out. We must find a way of going beyond the traditional scientific community, but how we engage. The stakeholders like society are a very big scientific lobby group, by the way, politically. So it's very, very important in your scientific careers to find a ways of getting to them. And again, because I've highlighted this before, at the very core of the open science recommendations, there's a pillar of opening up this dialogue with other knowledge systems. I happen to have been in the committee for the Director General Committee for the UNESCO recommendations and implementations. I always say, not just the dialogue, colleagues, we must do more in terms of advancing that area, especially in developing countries. You can see how that ties in with the fair and care principles and also with the bioeconomy. There were action items for countries in terms of all these things. Obviously, those who also translate to action items to universities, to institutions, and what needs to be done and to be reported every 4 years. And I think it's critical, even to individuals. We need to think about where we are. So have that value proposition one-pager. Tell your PI, tell your dean, tell your VC, tell your DDG. Sometimes they say fish rot from the head. We also need to educate those that are at the top. Why should I be opening up more of my science and spending more time on data creation and all these other outputs, not just on the paper that is cited on Scopus and why the research evaluation system at my university need to take that into account, colleagues. So it's very important. And obviously, I also generated this, be able to articulate, especially with the frameworks that we already have. There's worry that open science is also now degenerating in terms of the progress that needs to be done. But you can see the impact pathway is straightforward, and you can link it to the various open science components, and you are operating the data space. So for example, you can then push that with good data curation, the basis for data-driven decision-making is obvious. So these are, I'm sure when you do your proposals, I mean, donors now are very strict about obviously a lot of criteria. Surely, somewhere in your description, in your project proposal, you can find a way of throwing in some of these SDG arguments. I mentioned data-driven decision-making. We're at the UN as last year regarding this, and I think it's key. Some jurisdictions, I can see some Europeans here, you cannot manage what you cannot measure. The value proposition of open science, I think, is fairly evident now in terms of evidence in the scientific literature in areas like citizen science. But we must have impact framework, to do some things. Mission-oriented science, moving away from doing science business as usual to that. Important. The ISC, the International Science Council, came up with this framework. I believe a number of hubs have been appointed to drive this mission-oriented science. I've got 5 minutes. I think that's sufficient. This is critical. And of course, what the young lady was talking about, what is the end product? We know that both of these are important. Curiosity-based research and challenge-based research. How you balance that as a country depends where you are. If we're South Africa, maybe you need to think about the ratio, how much you invest in basic research and how much you invest in the other. If you are the US or you are Canada, you are Japan, it could be that you also think about that ratio. But nonetheless, the balance is important, but either way, there's an avenue through open science that we can connect the two. I don't want to touch this very thorny subject. You all know about it. It's the gremlin in the cupboard. It's important, especially for young career research. You don't want to go this route. Avoid the temptation. You know, I have a famous discussion with my brother. He's now a vet. When he was a second-year science student, he always talks about this story where he was doing titration. And he had this, I think it was his test tube was full of this substrate. There's, I think it's a salt, maybe some acid and something. And he looked at his friends, and theirs didn't have as much. So he threw half of his into the zinc, because everybody else's looked like it was there. As it turns out, his experiment was very successful. It's just that, you know, he decided to falsify his evidence, so to say, thinking that he can conform with the other. This is important, colleagues. And of course, we know about the AI, that we are all talking about. We all have to think about. I'm a computer scientist. I always cringe when industry people talk about AI sometimes. There's evidence that we are throwing a lot of money with diminishing returns. So we also need to think about that. And of course, you all know the limitations. If I were to stop now, it would be this. I'll spend 2 more minutes. As Africa, we must do this. You cannot join a ship that you do not know where it's going. When I say Africa, I also mean the developing world. We must introspect and certainly domesticate this conversation so that we own them. And we certainly must think about, even if everything else was good, we have all the money in the world, are we configured for success? Only if we do that, we can make those baby steps. Rome was not built a day. The pyramids were done thousands of years before. Yeah? It's not a race. It's a marathon. Got to get it right. Which is why the dream for me, and Nikki will agree, and all of you will agree, is to have this policy-aware environments that are federated and they have all these very good attributes around them. And they obviously have these sovereignty-preserving characteristics, and we can see the full. We know we've got gaps. We know we've got gaps. We know the opportunities, and we need, as organizations, institutions 2 minutes to where we are. Who is doing this checklist? If I were to give this to UCT, what score would you give yourself? Maybe your research office should do it. As an ecosystem in Africa, if I were to say Southern Africa or Africa, where are we right now today? You find that those characteristics are still there. You know, you've got well-meaning champions like Nikki and others in your communities, but we are not there yet. We know the things. Africa has got policies that are already providing us with the foundation for driving these things. And we're talking about regional integration around those dimensions. Science is one of them. So when we discuss what we discuss, let's have a value proposition of how we're using what we're doing to do integration. This is my last slide. I've got 20 more, but I'll stop here. AUSP, I'm not hosting your data, but I'm doing what I'm doing today. I think these conversations are important. We're a platform that wants to convene and coordinate interests and ideas in your institutions and resources. Somehow I do have this clout now. Maybe I have to get used to it. I get invited to very big forums in the UN and the AU. The good news is that the AU is now, with STISA, seeing the value that through all of us and through ASP, we've shown why it's important to have something like this. So we are having conversations around aligning this with the AU policy frameworks so that the Africa in the ASP makes sense. People are always asking, why are you African? What's your legitimacy? So now we will have some legitimacy because of STISA. And of course, we have all these good things. I'll stop here and say it's very promising that a lot of countries are doing their open science policies. When their implementation plans gets done, as a community, look at them and think about projects of national importance, of regional importance, of continental importance to make sure that we can leverage these endorsements with some practicalities. Certainly as ASP, we are now looking to engage around these implementation plans and make ourselves relevant. I'll share all the other slides for your indulgence later on. Thank you very much.
Thanks, Chiamo. That was really amazing. And you've given us so much to think about. And we'll take questions for you now. I think just two things that always resonate with me, and that's also sort of in the back of my mind when we, at these conferences and trying to understand how we move our work forward. And you spoke about incentivization of our work and how I think in academic institutions, we should incentivize data sharing within that career progression. And I'm not sure if that's happening yet, but I think you raised that as well. And then also in terms of the scientists taking on the role of advocacy, talking to its community, its institution, the scientific community, but then to the politicians and the people that actually make the changes. For me, maybe you can speak to this, like in South Africa, and I think scientists from South Africa can sort of recognize this or agree with me that there's a big gap between the scientists and the politician. We don't have that opportunity to speak to the politicians. In other countries, it's different. So that is a bit of a problem, and maybe you can speak to that. So I'd open the floor for questions for Chiamo.
For answers?
Answers, okay. I think that's what, yeah, first.
This one is at the end.
Thank you. Thank you so much for an excellent talk. And so I'm going to pick your brain. So I'm Dr. Ini Ogundeji, senior scientist from the Southern United States. So as I think through lessons learned over the course of my career, now working more on private-public partnership, I wonder if you might speculate, in an ideal world, what would a great private partner be able to assist in some of the processes for moving the needle in terms of, if we think about infrastructure, supercomputing capabilities, sharing, what would an ideal private partner look like in that scenario? Thank you.
Very nice. Arsenal fan, feel my pain. I lived in North London for like 15 years. I'm a supporter of what I'm doing. You want my comments? You want to hear about Arsenal? Okay. No, thank you very much for those two questions. I think for your second question, we need, somebody mentioned equity. You know, these equitable partnerships are key. My experience, I've worked in industry a little bit. I remember during my first year PhD, I was at the British Telecoms. Industry by their very nature, you know, are driven by shareholder. They've got to demonstrate value. And it's very competitive. And more often than not, you find that they're primarily driven by their own strategies that are approved, obviously, by their boards and everything. There must be, especially through maybe some vehicle of corporate social responsibility, to have an honest assessment of how they want to play a role in science for good, not science as a commodity, and how they want to apportion some of their investments for that. I mean, if you talk about, for example, infrastructure, not many countries in Africa can invest in supercomputing facilities. One of the big ones is right here in Cape Town, at the CHPC. The other is in Morocco. But we know about the huge data centers around the world. How can we work with the private sector? I talked about creating this federated policy aware research environments where we can do science that is meaningful to society. I would argue there's an argument where we can work with industry to really provide access to computational infrastructure for science. A lot of scientists like myself decided to come to Africa after 20 years, maybe away. You come back home, you cannot do your science. You have to talk to your PI maybe in Chicago to be able to access some little machine, some big machine, sorry. If I had time, I'll also show you the project that we did with the Texas Advanced Computing Center. Every 4 years, 3 years, they decommission their huge machines. We shipped some of their computational nodes and capacitated universities in the continent through the leadership of the CHPC Center here in South Africa. It's a form of relationships you can build to make it accessible for scientists to access infrastructure right in the continent. Of course, you can talk about the cloud. And I remember I was at University of Botswana, Microsoft was on our case around maybe moving on to their cloud. And you're going to be charged, I don't know how many cents per second. I'm thinking, I've got 500 students. How much is my bill going to be? And we're talking undergraduate. So that's a fantastic question. I think there must be somewhere in the big pile of money that they make that they can apportion maybe to some of these avenues. Obviously, I think in areas like bioeconomy or genomics, surely there's a value proposition where it could be a win-win for them. The same with things like climate change and environmental work and disaster risk reduction. There's avenues where we can engage. So with my little industry experience, I think they will not lose by doing this. They will always win. I remember also sitting in a board in a state that's a government-owned entity that was leveraging technology, and they were always wanting to use these proprietary technologies. I said, work with the ecosystem to develop some technologies. So it's a good question. I don't know if I'm the right person to answer it, but that's what I would do. With yours, you must have scientists wearing suits on prime time in prime television in SABC talking about this. Where is the SABC cameras? So scientists must talk. I spent 2 years of my fledgling career seconded to government to help with some of the digital transformation initiatives that were there. No papers for 2 years, but I think out of that, we developed the Botswana National Research and Education System network. This after 10 years of IDW, the conference I talked about. Eventually, Tiamo, you used to make noise about this. Come and help us do it. They say, yes, we'll come and do it. So we have to make ourselves available for some of these things. Thanks, colleagues.
Thank you so much, Dr. Samo. Impressive presentation. So I really enjoyed the fact that you zoomed out from, like, let's say, small vision of a researcher to a bigger holistic and strategic view regarding curation, data, dealing with data. So now my question, we all know there is a huge amount of work we are facing for established open science and applying all these principles, care one, and the new ones to be implemented. So my question is, what's your thoughts regarding contacting new initiatives and all one nationally and continental scale regarding genomics. There are, I mean, there is multiple initiatives, and how to be. I mean, you are attending very, let's say, stakeholders level conference to convince them to implement those principles. And as you may know, so implementing at the top of the hierarchy could give more and more push to implement those than start at the bottom as we researchers. Thank you.
No, I think your answer is as good as mine. I mentioned the South African policy that has been now approved. I think if governments can, obviously, they have STI policies, they can then also develop open science policies whose implementation plans must talk to, for example, strategic projects. For example, if genomics, as we all know, is a very important area, there is a flagship project in South Africa and in Africa that we can use as a conduit for advancing these principles. That already gives us an avenue for coagulating and connecting the communities that are already doing very good work to coalesce and create the necessary critical masses and the collaborations needed to do it. I'm not saying governments can impose, but I'm saying they can provide an environment where through the implementation of the policy frameworks, they can get communities to work together. I don't know about Nikki, how you find yourself working with other networks outside your own networks in the continent. Sometimes it's a personality issue, whether someone likes Nikki and they want to do their own thing because some funder wanted to work with that community. Funders must come to us. When I say us, I mean the African countries, African funders of research, when we have our own research agenda and they join it so that they can consolidate in terms of what we do. So the issue that I see personally is that there's a lot of funding pushed by funders driving their own agendas, and we don't have a coherent national, continental, and regional agendas around which already we can coalesce. So long answer short, I think these policies with strategic use cases. I mean, for example, STISA talks about health, agriculture, and others, and they're saying there could be some flagship projects there. So I think if these things are implemented under the PRISM policy frameworks, that gives us a better chance to be able to work together. Thanks.
Hi. Hi.
Oh, did you want to do?
Maybe for, sorry.
Oh, sorry.
Can someone give me answers?
Can she go next? You?
Yeah.
You can go.
Okay, sorry. Thank you for your very interesting presentation. So my question is, according to UNESCO guidelines on open sciences, what would you identify as the most urgent priority or the primary strategic priority for advancing and supporting biocuration capacity in African scientific community?
No, I'll go beyond just your community. I think there's something in industry called change management, isn't it? It's in the mind. How do we change our way of thinking to make sure that we achieve what we need to achieve amidst all the other factors? I personally think it also, one of the key things is the incentives to be able to incentivize us to do what we do. If we all agree that at the very core, it's all about the data, primarily, of course, and other things. How do you produce these good quality data sets as biocurators in your domains? And how do you incentivize people to do that more and more and recognize the value of what they're doing to make sure that we can invest in those skills as we talked about? So if you were to ask me, policy and incentives, critical. Of course, infrastructure and other things are enablers, but we need to change the people, and we also need to change the policies that direct what we do. I think we can start there. You should be rewarded as a data steward for the work that you do, handsomely. I'm not saying just monetarily, but also in terms of career progression for your contributions too. How do you support? Infrastructure. Yeah, infrastructures. I mean, you are from the US. The money that NSF is putting into these big supercomputing facilities sustainably over a very long period of time. It's a strategic investment. Doesn't mean that it's cheap, but they've recognized this as a strategic investment. People are now talking about quantum computing, very expensive, but we know its implications to security and cybersecurity. I think countries need to think about where science fits in the grand scheme of things. And I think it's what our politicians are elected to do, to be able to weigh these strategic investments and see, for example, as South Africa, do they continue funding the HPC environment for the next 20 years, or they want to stop, or as Tunisia or as Morocco. I went to Morocco to see their fantastic supercomputing facility in Biocuration. I think obviously, you know, there's good money with the phosphate that Morocco is producing, and they've invested it. And you saw that UM6P campus in very good facilities. And I think with those type of investments, not everybody can do that. But if we do that, maybe let's have modalities for allowing the continent to create these clouds that will allow others to access, maybe with some modalities. Not everybody has to have a supercomputer, but we can create an environment where it can become sustainable for those that invest, to make sure that they are fully utilized there. We can take on. I think I asked her a question. Maybe she can.
Thank you so much, Dr. Shamo, for that presentation. Right from the first day that we spoke after my presentation, your career has been something that is something I teach my students about, and it's so nice to meet someone in person who is actually living that life. And it's not always common to actually find people who love their jobs. And you started this presentation with, you have the best job in the world. Yeah. So I would like to ask, you seem to know about almost everything that we do in the sciences. I'd like to ask, what has been the most challenging part of your career so far? And secondly, I would also like to ask, there's a recurring advice or suggestion that has been given by senior scientists here, Prof. Nikki, Dr. Peter, and the likes, about networking, community, and mentorship. So I'd like to ask if you have anything in addition to add to that for the young scientists here. Thank you.
No, look, that's a very important question. I mean, to ask me what's more challenging, which scientist does not want to do science? I look at you and enthusiasm, and I'm thinking, 2001 or so, my first year PhD, direct from my undergrad with a very good first class, brilliant, string of A's, 100%. I wanted to do physics, I wanted to do chemistry. My chemistry was fantastic, by the way, top of the class. And I ended up saying I want to do computer science. 1996, I got a scholarship. When I finished my undergrad, my professor, that gentleman, said, Tiamo, you must do a PhD. This is after I spent a year on an industrial placement in a nuclear power station in Hisham. I said, I'm not so sure. He said, go think about it. And he managed to get me a scholarship into the PhD program. After that, you're thinking, all right, science, you come to Africa, you see the environment is not good to actually do meaningful science. So those little sacrifices that I think we must do. Maybe my son will be a better scientist, I don't know. So that's my only regret. I was trained as a scientist, but I'm now wearing a suit. It's a big sacrifice, but you need to think about that. Mentorship is important. I mean, you just sitting next to Nikki, something is happening there. You can have a virtual conference. Let's sponsor students to go to international conferences. You should have benefited a lot more by going to that FDO conference than me in the twilight of my career, because then he's got a full life ahead of him to enthuse about these things. The same with the International Data Week. I want to see an African conference with African representations, especially with postdocs, so that they can have the torch, be the torch bearers going forward. Don't get me philosophical, but that's what I would say. Thank you. I think we need to create a mentorship network. Mentors are busy. I don't know how much time you have, Nikki. I have undergrads. You know, when I first came as a postdoc coming to University of Botswana, I taught 400 students. First year, principles of programming. Second year, object-oriented programming. Third year, systems programming, masters, and I had a PhD student. How much time have you got? Today, I have emails. Say, can you write me a reference for my job? Which one are you? So write me some, but it's important. Sometimes I get my hand shaking by this guy is big, you changed my life. I said, how? Who are you? But it's a legacy, isn't it? Where Nikki and others are doing a fantastic job. We must have a professional face, smile, and give the little tap that we have to others. I think the keynote in the first day about networking is important. You can see I still think highly of my supervisor, even though he abandoned me in my second year. He threw me in there, but then I had to sweep. This thing, oh, my supervisor think for me. No. After your second year, you are on your own and your network. And you need to start thinking about how you mobilize your resource. Thank you.
Thank you so much for the questions. And on behalf of, thank you, Chiamo. On behalf of the organizers, we'd like to hand you another torch. Okay, here you go.
Thank you very much.
Please take a picture. I think I'm.
Normally, as a politician, I have to open it to show you what's inside, but I trust it.
Thank you so much for that. And thank you for all the questions. And I'm going to hand over to my co-chair now. Thank you.
Thank you again, Dr. Chiano, for this great plenary session. Now we are moving to session number 9, which about ethics, data, and governance. So our first talk will be given by Professor Nikki Tiffin from South African National Bioinformatics Institute, South Africa. Her presentation will be about from principle to practice, ADBX tools for equitable data and biospecimen sharing. Professor Nikki, I invite you to join us, and thank you. The floor is yours.
Thanks very much. It's really a pleasure to be here. Thanks for the invitation. Yeah, so I'm talking about the ADBX tools for equitable data and biospecimen sharing in 10 minutes. It's going to be a bit of a canter through the material, but I do urge you to go and look afterwards at the tools that we've developed for some more detail. So yeah, the principles of data governance, I share this framework quite often, and I'm not going to get into the details. We really have these important principles that are about ensuring maximal use of data to improve lives, but also doing no harm, representing underrepresented populations, making sure we distribute benefits where they're due, and really being intentional about our governance and our governance frameworks. So in practice, what we do in my group is we develop tools to try and make it easier, because we think researchers don't really have time to go and read through all these principle documents, however important they are, or to dig into the guidelines. So we try and make tools to make it more understandable, easier to use, things that are functional, and we're really supporting equitable practice and also stakeholder relevance. And I want to say a bit more about stakeholder relevance, because I think it's something that we tend to forget. So I'm a researcher, I'm an academic researcher, and I recognize that as researchers, we tend to get this research tunnel vision about data. And it's kind of unhelpful when we're talking about data governance in a broader picture. And we've just heard also from our great keynote speaker that there are more stakeholders in this game than researchers. We also have to think about the commercial use of data. This is very much at the forefront at the moment with the PABS negotiations around producing products from data and how we can do that more equitably. And then public health is also a whole other space for data use. And that's about providing health services, it's about health policy. And then researchers, we tend to think about generating this evidence base, publishing what we find. So yeah, open access data, that's really about research, and it's not always entirely relevant for the other use cases. So yeah, I also see a lot, I mean, we've got the open access principles, the FAIR principles, and these things all revolve around getting data for research, ensuring access to end users, enforced by funders and journals, and data access committees who are all about getting data to the end user. And the FAIR principles also about getting the data, the end user, and making it easy for the end user to get these data. There's been an attempt to add the E But I've done a lot of research and I can't find the origin of the E beyond a WHO footnote, and nor have I found anything about whether it stands for ethical, equitable engagement. It seems to be a bit of virtue signaling, and I hope someone can point me to the substance behind it, because I'd like to explore it further. So I'm going to tell you about two tools we developed. The first is the African Data and Biospecimen Exchange. It's live, it's up and running. And this is a platform for sharing data and biospecimens from Africa. It's a listing or a catalog where you can catalog what you have, but you don't have to send anything in, nothing centralized. It's not a repository. It's a low barrier to entry in that the listings are quite easy to populate. There are no like very strict rules about how you describe what you have. And then there's a chat function. So if you're sitting outside Africa and you want to use some African data or from within Africa and you see a listing you like, you can contact the people who have put that listing up there, have a chat, negotiate how you're going to use the data or the biospecimens and come up with a fair deal. And once you've concluded the chat, there's the inbuilt functionality to create your sharing agreement. So it's like, I mean, I always date myself. I call it like a wizard. It prompts you to help you get all the right clauses into that sharing agreement. And you might then take that sharing agreement onward to your legal department to create a legally relevant document. But everything you need to know about your sharing agreement, you can put together with your end user or the provider of the data or samples through the site. And of course, this gives you the provenance because everything's stored there, your chat, your agreements, how you're going to go about the sharing. So it's live, it's up and running. People are uploading listings of their data and biospecimens. We are working on some additional functionality, like building APIs, bulk upload facilities for the big biobanks and the big data repositories. And this is just to show you what using it might look like, just reiterating what I've already told you. If you're a resource generator sitting in Africa, you can it's all by profile identified users. So you create a profile, which is very quick to do. You can use your Orchid ID or your Google ID or create something bespoke and list what you're holding. I've got a data set that looks like this. I've got a sample collection that looks like that. And you can say how you're willing to share it. So we've got lots of modality in there, federated analysis, TRE, collaborative only, or traditional direct sharing. Someone who's looking for that, the resources consumer, will also create the profile, get online, search and discover the resources they want to use. You connect with the chat, agree how you're going to do this sharing, what the benefit sharing is, if that's part of the agreement, and build your sharing agreement online. It looks like this. This is the site. As you can see, you can log in, sign up. You can't transact unless you're signed in. So if you're just having a browse and you can't understand why the clicks don't work, just create your profile and log in and you'll be able to transact on the site. So yeah, it's about changing the dynamics of how we share and giving a bit more equity and a bit more autonomy to the Africans who are holding data and biospecimens. What it is not, it's not a place to upload data sets or send in samples. You don't do that. Once you've negotiated how you're sharing in that space, you also agree how you're going to do the sharing. And only when it's all done and dusted and signed, you would send your resources off or receive the resources. It's not a replacement for other repositories and biobanks. It's complementary, and it's a way to advertise and share what you have. So your data can be somewhere else, but you can still list them here. And it's another way of exposing to the world what it is that you're holding. So we're not trying to replace anything or to supersede anything. It's another tool on top of that. And it's not a mechanism for transferring data or specimens. You would only do that after you've agreed how to do it. So we've also got a second product under the same ADBEX umbrella, but whereas the platform for sharing is something for Africans to present what they have to the world, the data sharing license is something that is for everyone. It's actually for any kind of data, anywhere in the world, public health, commercial, research use. It's not restricted to our world of sort of bioinformatics or biocuration. And yeah, this is also live, up and running. So I've put a Creative Commons little badge in the top right there. I'm sure many of you are familiar with Creative Commons licensing, but you probably haven't found exactly what you need for data sets. So we've created something that is similar to Creative Commons, but it is specifically designed for sharing data sets. So if you have a data set, you're willing to share it, and you want to clearly communicate your terms and conditions, you can use this tool. It's very quick and easy. It's intuitive. I'll show you the landing page in a minute. And you can answer a series of about 7 questions about your terms and conditions. And once you have filled that in, you'll see the license building on the right. We've included T's and C's around attribution, the types of sharing, what publication is allowed, how you can disseminate your results from the data. Specifically benefit sharing, because we really want this to push equity and more equitable practices. So if you want some kind of benefit sharing, you can record that in your license. Of course, not obligatory. It's up to you what your T's and C's are. Whether commercial use is allowed, cost recovery, onward sharing, whether it's permitted, and then share alike and derivatives, similar to the Creative Commons kind of approach. This is the landing page. I think it's kind of pretty. I really have enjoyed building this project. It's been something we've been planning for a long time, and I was able to actually implement it with a fellowship with the WHO and in Berlin at the hub, and work with them to put this together, because of course it's very topical for sharing data in a pandemic or similar kind of in the pathogen genomics world as well. So yeah, please visit, give it a try. We don't store any data at the back end. You create your license, you download your icon, your PDF. We've got machine readable code. There's JSON codes if you want to pull it into a database. We've also got a code for each type of license. Yeah, so please give it a play, and we'd love to have feedback as well and enjoy it. It's something that we've built in Africa, but for the rest of the world. So yeah, I hope you get something out of it. And just to say thanks to everyone who's funded this. The work was funded by a Calesta Stuma Fellowship that I have from the Gates Foundation. And Hominem and Method Lab are our fantastic partners who've done the hands-on dev. They've done a fantastic job. So yeah. And then, of course, my team, Tana and Eddie, were on this project from the start. And yeah, I've been very lucky to work with great people. So thanks very much.
Thank you very much, Professor Nikki, for this insightful presentation. If you have questions, please take note. We will go with them in the discussion session. Now we are going to the next presentation, which will be presented by Dr. Ulrich from Heidelberg Institute for Theoretical Study, Germany. Her presentation titled Enabling Reproducible and Searchable Research with FAIRDOM-seq and FAIRDOM-hub. Please, the stage is yours.
Thank you very much. So yeah, my presentation will be about our data management platform, FAIRDOM-seq, FAIRDOM-hub. So how to enable reproducible and shareable research. So first, I want to point to a survey which was done about 10 years ago, published in Nature, where researchers were asked how well, or I would ask you, how well is your experiment documented? And researchers were asked if they have failed to reproduce experiments. So either their own or also other kind of experiments. And as you can see here, so it's independent of chemistry or biology. So most of the people, it's above 60%, were not able to reproduce experiments from somebody else, and also not their own experiments. And then the question is, so why, or what could be the reason for that? So of course there are different kind of reasons, but what's important in our case for data management is that methods, codes is unavailable, and also the raw data are often not available. And that's one major problem to reproduce an experiment. With that, I would like to point to the research data life cycle. So this cycle is taken from the IDM kit, which is developed within the Elixir framework, reporting or giving guidelines for research data management. And the problem, most of our experiments is that we are planning, we are collecting, so we are planning our experiment, we are collecting the data, we're processing, we're analyzing the data, maybe we preserve that, but then there is often this gap of sharing and reusing the data. And so that's why we developed, or it's not why that, but we developed this research data management platform about 20 years ago, together with partners in UK and Belgium and Norway and Netherlands and in South Africa. So there is a software which is called Seek, and this is an open source web platform to store or catalog different data types. There are no file format restrictions. You can structure your data based on an ISAR standard for investigation study essays. So you can define your experiments, aggregate it into studies and investigations. You have flexible, detailed sharing permissions. You can assign UIs for individual items or also to the entire data set. And of course, there's also an API to automatically access and submit the data. And here you can see what kind of data are stored there. So we have people data. So for example, something like yellow pages that you can see who's involved in which projects. We have data files, we have samples, protocols, workflows, presentations. You can group everything for an event. You can link your data with already published papers, and everything is connected to a project or program. So here you can see how these data can be interconnected. So everything is grouped in a project. And then you can have your investigation study essays to define, for example, smaller work packages within your project and specific studies, and this essay is a specific experiment. And then you can link all this data together to these experiments, so your data file connected with a model, with a document, with a protocol. So this is an example of our collaboration partner here in Stellenbosch. So they, for example, have an investigation and connected a lot of studies to that and a lot of experiments and different essays. And for each experiment, they connect a data file, which is an Excel sheet. They connect a model because they are systems biology experts. They have a modeling tools and they develop some models for biochemical pathways. And then they have the protocol connected to that. So one other example is to have samples registered in the platform. So you can define different attributes for your samples, so comprehensive metadata for your sample type, and then you can generate an Excel sheet and upload a set of data files directly via an Excel sheet, so you don't have to put all samples separately. And you can see there experiments where people connected hundreds of samples to a specific data file. For example, you have your Excel sheet and then you have the corresponding samples connected to that. Also for other kind of assets, you can define your own metadata. So we have some basic metadata like the title, the person who uploaded that, time and description and things like that, also licensing and sharing definition. But you can also define within a different project, with a specific project, you can define your own metadata. So for example, if you are working in a plant specific project, maybe then you have specific requirements for your project, and you can also define that here. And people within your project, they have to follow this metadata. So they have to, for example, or you can define what kind of metadata they have to submit, and which are and others are maybe optional. what's also important is to have a tracking of different versions. So, for example, if you have a data file and you have some changes within the data file, then you would not change that within the system itself, but you would upload a new version of this data file so that you can track over the years or over the project how the data files evolve. And everything is connected to the original data file, for example. Most important for our users is, of course, the data sharing. And here it's important to say that you can keep your data private, so you don't have to share it, you don't have to publish it, but you can also share the data with different person, with different project, institution, or also within a project or program. And here you can see, so for example, I have a data file, for example, and so the public is not able to see that, but people within a different working group, within a specific project, they are able to view, download, and others are only able to view it. But for example, my colleague, she is able to also edit and manage this data file. So for that, I can really change also during this project. I can also change this sharing, so I don't have to do it from the beginning. I can also switch these access rules during the project phase, for example. And for example, if I want to share it with the public, then I can just switch this button here, and then now the public is also able to view and download that. And so all non-registered users of this platform would be then able to see and download all the data. And if I have published data sets, then I can make a snapshot of it. So for example, here I have such an ISA tree, so investigation study assay, or my data are connected here in the tree-like structure, and I want to publish that, and then I make a snapshot of it, and then I assign a DOI for this whole snapshot. And I can continue working on that, of course, it's not frozen, but this one, which is assigned a DOI, this state is frozen for the DOI, for the publication. And the advantage of that is that you can use it as supplementary material. You can just use this single link, this single DOI, to refer to your whole data set. So for example, this example here from our partners, they have assigned a DOI in their publication. So they said the data are available via this DOI. And if you would click on this DOI, you would get all these data files, models, SOPs, studies, essays, everything, and you keep this tree-like structure. So it's not like your normal supplementary material where you have one PDF containing some tables, some figures, some text, some protocol information, and here you have that structured based on your experimental structure. So the FAIRDM-seq software is an open source platform, so everybody is free to download the software and also install their own instance. We have one instance hosted in our institute, which is called FAIRDM Hub. It's free to use. Everybody can go there and register and then upload your data. So you can use it for your PhD thesis, but you can also use it for your collaborative project. And of course, the advantage of having this public instance is that you don't have to care about updates and backups and things like that. This is just to show you that there are other projects having their own instance. So if you want to use that, you can either go there to our FAIRDM website or see who is doing similar things. So they, for example, also develop their own features, which sometimes also were included in the main branch of the seq development. This is our team behind. So we are at HITS in Heidelberg. There are people in Manchester, at the University of Manchester, people in Belgium, at the MIT in USA, Amsterdam, Stellenbosch University, Edinburgh. So there's a huge group behind that. So if you want to be involved, you can also go to these websites or use the software. Feel free to also contact us for questions. Thanks a lot.
Thanks so much, Ulrika, and I'm sure lots of you'll have lots of questions during the discussion time. So I'd like to take this time now to invite Dr. Guy Cochrane from the EBI, UK, for his presentation on biodata infrastructure towards global contribution and global benefit. So welcome. Just to say that we don't have the next speaker, and Dr. Njangwi Munang won't be joining us. So we'll go after that into the panel discussion. Thanks so much.
Right, well, thank you very much. And thanks very much to the organizers. It's great to be back at ISB, back at Biocuration. So if you are a life scientist or a biomedical scientist working anywhere and wherever you're working, you are quite often producing some data, and you're almost certainly always consuming data that are being produced by other people. And to do that, you transact your data, you push your data, and you pull your data through databases. And there exist lots of useful databases, but there's a core of very, very heavily used databases that I'm going to focus on a little bit for the next few minutes. So if you look at the map on the left, then you just work out one way. There we go. If you look at the map on the left, then what we're looking at here is for a core. I'll come on to define what I mean by core, but we're looking at the usage of databases. And if you look, so this is citations, we're looking at citations of accessions and identifiers for records in databases. So these are data citations. And there are very few countries where you don't see, where they don't light up. So there are very few countries where people aren't accessing data at all. Now, of course, because in different countries, there are very different scales of scientific activity, the rates are different. So it's on a scale. But the important thing is that there is access everywhere. And if you're doing science, you need access to these resources. If you look at the map on the right, though, you see quite a different picture. So this is looking at where the databases operate, who is operating the databases. And you see that the vast majority are operated from North America, from Europe, and a few of them are coming from Asian countries. So there's an imbalance here. And I think this imbalance is problematic. And I think the world would be better if we redress this balance somewhat. So I would argue that a greater participation, a broader participation in actually operating the data resources, the databases, the infrastructure is really important. So a biodata infrastructure for all, by all. And there are three drivers, three reasons I think this is important. One is for the sake of the sustainability of the databases themselves. The second is to increase the quality and the value that are offered by the databases. And the third is to help to increase global equity. So for the scientists involved in working around the databases and working and using them, but also perhaps more broadly for citizens in general. So I'm going to step through these things in a few minutes, but a few other things to say first. And really this BIOL, so what kind of participation am I talking about? So there's already, the value of these databases actually comes from a lot of different places. Informally, the biocuration activities, data stewardship, ontologies, software, workflows, working on data standards, all of these things contribute to the value that they offer. It's a collective effort already. ISB has been really important in focusing on biocuration, and I think giving it stating that this is a profession, it's in itself, and it's an important contribution, and I think ISB will continue to be important. There are attribution tools. Perhaps in some areas there's better attribution practice, but still we're a long way to go before actually biocuration contributions, ontology contributions, software contributions are fully credited in the way that I believe they should be. I'm not going to focus on these. I'll call these informal contributions. I'm not going to focus on these for the moment. I'm going to focus more on the second kind of contribution, which is much more formal, where an institution actually participates in an international collaboration to deliver a database or to deliver a data resource. So many of the existing international databases, the global databases, have an established pathway for institutions to join, so for new members to join. So the sequence databases have this, the structural databases have this, protein interactions, proteomes, taxonomy, biodiversity. So there is a formal path that institutions can take to join and to participate. And those sometimes they're symmetric arrangements. where everyone does a share of the overall job. Sometimes they're asymmetric, where one party contributes one component. So maybe does the biocuration, or does the training, or does the software development, or so on. So back to walk through these drivers. So sustainability. So I'll step out again because this gives me a chance to introduce where I'm coming from. So the organization I'm representing is the Global Biodata Coalition, which is a coalition of life science, biomedical, biodiversity funders that come together. They recognize the importance for their research programs of these databases. They realize that their research programs will suffer if these databases fail or become less useful. But they also understand that the system is very fragile. These databases struggle for sufficient funding. They struggle for funding of the right, for funding with gives them long-term strategic planning abilities, and they're actually at risk. And so it's a very fragile system that the world is depending upon, and the funders realize this. And then they agree to work together in different ways to come up with better international cooperative methods or models for funders to work together to support the data resources and to improve the situation. And so we have 11 members. I know the African Academy of Sciences as a member for some years now, and we have an observer in the South African Medical Research Council, which is hopefully on a path at some point to membership. And then our work is underpinned by a grant from the European Union, and this runs the coordination activities that are hosted at the European Bioinformatics Institute. So I can't cover all our activities, but one of the things we do is to run a program for what we call the global core biodata resources. So it's a selection. We have an external expert review. People who run databases, they submit. There's a set of defined criteria, and there's a review against those criteria. These databases work closely with the GBC, and it's with these databases that we'll be trying to build better solutions and we'll be evaluating and so on. And so we've had mention of many of these already. You'll notice in there. So we've heard about DDBJ, we've heard about Interpro, we've heard about disease ontology, we've heard about HGNC, we've heard about Reactome, Kemble, probably many others as well. So these are databases that hopefully are familiar to lots of people. So the reason having greater participation, broader global participation in running those kind of data resources adds to sustainability is because it brings in additional people who can contribute to the scaling of the whole effort, because everything grows, as we know. But it also typically brings in different funders. So in many of these existing collaborations, that you have completely independent funding. So in the case of the sequence databases, you have 3 different sources of funding. And if one fails, which we hope it won't, but if one fails, then there's resilience in the system. Now, the broader the network of providers, then the greater the resilience. So having more resilience in the system makes something a lot more sustainable. So I'll move on to quality. So if you have more participation in the system, you have more energy to ingest data, to curate data, to process, to build services, and so on. So you end up doing a deeper or perhaps a broader job. One thing that is complex to deliver from a service provider perspective is really tune services to different parts of the world when you're not representing those parts of the world. So, knowing about the science that matters in a particular region, being in the time zone, speaking the language, understanding how science works in a region or in a country is really important. So the quality of the data that come into a system, the quality of the curation, the value of the overall system can increase if there's broader participation. So finally, onto equity. So if you have a very biased representation participation, then you end up focusing on the things, of course, that matter to the countries that are participating. And so we've heard at this meeting quite a bit about underrepresentation of African genetics data or genomics data when looking at human populations. There's also an underrepresentation of many from different parts of the world, many unique biodiversities that are not covered well. Of course, there are different disease burdens, different infectious disease burdens that are more important in some parts of the world than others. And of course, they're important agricultural species. So there's a lot of science that is regional and local and national that doesn't get the attention it would unless there's some participation. And so I spend There's a lot of tension as well, as we heard earlier this morning, between data sovereignty and how open science is practiced. I spend a lot of time at the PABS discussion in Geneva, the WHO discussion about how to balance these things. And it's tense. There's often a standoff between people who say, no, it must be very closed, because that's the only way you protect the rights and the sovereignty, and others who say otherwise. The tone of those discussions, if you had on the African continent, a node participating in the sequence databases, or in South America, if that was the system for doing data sharing, then the tone of those discussions would be completely different, I think, because it's an opportunity to not be consulted and asked how to do something, but it's actually making the decisions. And if you join, for example, the INSDC as a member, you become an equal member to the existing members. So you're sitting and steering. You're making decisions. You're not sort of responding to what's available. So just to finish, at the GBC, the next important part of our program is to start to pilot new models for building sustainability into databases. We have a number of different themes we're looking at, but one set of pilots we want to run is we call distributed services. So this is where you take opportunities to build broader participation into the operation and the running of the data infrastructure by in some way inviting or encouraging or doing things that help new partnerships to form. So these may take, we haven't structured these in particular, so we're sort of at the point where we're working out what the shape of these will be, but very interested to hear from people who are working in and around some of the global core resources, or maybe other data resources, who feel they have an opportunity here to get more involved. And of course, we're talking to the GCBRs. So hopefully we will be able to pilot and evaluate some pilots that will demonstrate some of the things that I've been talking about. So with that, I will give you a slide, some pointers, and do sign the open letter that talks about the value of databases, and do get in touch if there's I think, well, we have a question and answer session in a moment, but do get in touch. I'm here for the rest of the day, and there's an email there. We're very keen to talk to people who are interested in using databases and their sustainability. So thank you very, very much.
Thanks so much, Guy. So I'd welcome Ulrika and Nikki to the panel, please. Make yourselves comfortable. So we'll stick to the 20 minutes given for the session. And yes, I'll welcome questions for our speakers from the floor. I think I have a hand here. First one.
So yeah, great talks. This question, I'm not sure any of you may wish to sort of talk about these topics. So open science is really important, but it feels like there are some increasing risks to open science which are coming onto the horizon right now, partly driven by sort of the pace of change in AI and our lack of ability to perhaps put regulations and so on in place. Also, it feels like we had this open access era, and looking at the open access statistics, the amount of CC BY publications is actually going down now, and they're increasingly publications are CC BY NDD or NC, so non-commercial, non-derivative, which seems to be driven, my view is a profit motive by the publishers to try and get some of this sort of money from AI companies. So, and then another aspect is sort of the AI security risks. We're hearing that AI could be getting very good at hacking websites, and I fear that source code, people might start closing their source code because they're worried about the risks. So lots of risks. I wonder if you have a sort of any viewpoints on any of these issues.
Thanks so much. Anyone want to take that?
Sure, I can jump in. Yeah, I think it's a great point, and I think there are lots of risks, but I think another factor that's very important and which I alluded to is that actually sometimes the open access is not appropriate. And I can say that, for example, we work with data from health records and the data are anonymized, but the condition that we have that we're allowed to use those data is that we don't share them onward and don't make them public. And that's appropriate because there's no informed consent from healthcare clients to use those data. So I don't think it's only risk that drives this more nuanced approach. And then, of course, I mean, speaking as someone from Africa, from the global south, we got very, very burned in the COVID pandemic because we were very open with our data, and I would say the global north were not very open with their products in return. So at some point, it becomes actually a self-protective mechanism because we don't have a trust-based relationship with the global north. And it would be great to have that, but it would be naive to say that we have it. So I think social equity is fully in practice. There are also going to be issues around open access. And I think inequity rather than risk is driving a lot of the reluctance to share as well. So yeah, I mean, a lot of people say, you know, there was this whole idea that we must build trust and that will facilitate sharing. But I actually think we need to build accountability and that will facilitate sharing. I think trust, and that bridge has been burned, unfortunately. So yeah, I think it's more nuanced than just risk that is driving this lack of open access. I think there are many more factors that drive it, and a lot of them are seen from the global south perspective. And I don't know that the global north has really been able to experience that lens on the way things work. Thanks.
Thanks so much, Nikki. Just before, Guy, do you want to add anything to that? Because, you know, around the infrastructure.
Well, I would just, yeah, I would just say I'm not sure we should look at the publishers for the trends on what's open and what's not open. I suspect that sheer numbers, actually more and more is becoming open because everything grows. But I wouldn't, I mean, as you suggest, the publishing industry has a sort of an agenda as well. So that may skew the sort of real attraction of open science or how much people value open science. I'd make a comment as well on the AI risks. I think so. The GBC and running its core program and the data resources within that, for them, this is a real risk and it requires a lot of work and a lot of thinking. And so the GBC sees this as another sustainability challenge. It's not just that they have to stand still; they have to move to do all sorts of additional things. There are many opportunities, of course, and as you told us the other day, I mean, there's an awful lot that AI can give, but there are some real hills to climb to make sure that it's doing that in a way that's safe and reliable and adds value rather than just damages things.
I'm just also wondering, just a thought that popped into my mind, would it be a good idea to have a conversation with these AI companies actually about, you know, the risks that we face as scientists in the repositories and so on? Maybe that's something to think about. So I'll go to the next question, Malik.
Thank you, Michelle. So I have a comment and a question. So thank you so much for the speakers. So we've been through this conference, which was a lot of tools developed. So my comment is regarding maybe effort regarding promoting and advertising your tools. It would be very interesting during this conference to see a lot of new tools that everybody is not aware about it. And my question is for Dr. Guy regarding the Bioporto Coalition. So during this conference, I'm sure that you'll be around some presentation developed through African initiatives. So there's any chance or there is any thoughts that you have that your coalition or your initiative can support all these developed tools for Africa and from African people aspect?
Yeah, thanks for the question. Yeah, no, so there is. I've been here, and there's a lot of exciting things going on. I think there's a real opportunity. So, I mean, with the pilot program that we're going through, but I think the bigger than that, there is an opportunity to work together through partnership on an equal footing. To have some of these discussions, and so there are real challenges, there are real tensions here. But I think that if perhaps through ABI or other structure, there were a node of the sequence databases or the protein structure databases, I think, and then that discussion would be happening within the databases. So the decisions on those discussions would be made by everybody. And so we'd have that discussion. And so I just think it would take a completely different tone. So I think, and then other activities that are around the data resources tools and curation programs and certain data sets and projects and so on, they can also plug into all of that. But I think if there was more of a partnership, I think we could have a level conversation as equal partners at the beginning, rather than coming to things when things are beginning to honestly fall apart a bit, and people have different things about how to go forward.
Thank you.
Thank you. Well, the questions for all of you, I know Dr. Cochrane's work is most closely connected, but I think that this environment that we have grown accustomed to of open by default is under threat. And so we have to go beyond the open science framework to try to think about some strategies for resilience and some activism. And so I was wondering if you could share, all of you, some of your thoughts on what could those strategies look like so that we can build more resilient resources, so that we can preserve, protect persons, protect data, while maintaining an environment that is as open as possible, but as closed as necessary.
Go ahead.
So I think if someone could build a kind of data sharing license, that might be really helpful. But it's the point is that I think there's clear communication of the terms and conditions of use, which is what we're trying to build with that. I think that takes away a lot of the uncertainty. We started in phage, we built a microbial data sharing accord, which some of you may be familiar with. And the idea around that was that nobody really knew what the baseline common decency rules are for sharing microbial data. So we started there as a sort of baseline. This is okay, this is not okay. But then with the data sharing license, it's taking it one more step. And that's saying, when I share my data set with you, this is the deal. It's unequivocal, it's clearly spelled out, there's no confusion. And I think that that's where accountability comes in. And with accountability, people are more confident to share. So yeah, personally, that's kind of why we built the data sharing licenses, to answer exactly the issue that you're raising. Thanks.
Go ahead.
Yes. I guess the question for all of you again. We saw in the US now a critical attack on some data resources. I'm thinking PubMed, or I'm thinking NCBI, when you suddenly go there and it says this is currently not funded and we cannot maintain the service maybe. So a question to all of you is, how do you think we can guarantee resilience of an ever-increasing number of data resources and tools?
Thanks so much. Ulrike, would you like to?
Yeah, I think a good example is this exchange, this distributed system of the genome databases, so that we have all the data in Japan, we have them in UK, and we have them in the US. So if some country would have a crisis, like maybe we have in the US at the moment, then we would have this data available. So maybe this is not possible for all databases, because it's smaller databases maybe would not work, but to have this kind of distributed system, and Guy also mentioned this, to include also African or Indian databases and into this genome data exchange, and to have that also on another level. I think that would be a solution to be on the safe side if something happens on one side.
Thanks.
Yeah, no, I agree absolutely. I mean, so no single point of failure. This is the problem, and actually, things happened outside the US, because the US has traditionally funded quite a lot of databases, outside the US. And so that was generous, but of course, every country has the right to make its political decisions, and no other country can, expect to change that. And whether you agree or not, you just shouldn't be relying on one. So having more funders in the mix, I think, for any activity, and probably distributing data and that kind of thing. But I think to your other point of your comment, I think we have to be ready to switch things off as well. So I don't think we should necessarily have always have proliferation of databases, or certainly not the big, really expensive databases. Of course, there are new data types and new domains of science that need to be addressed, so new functionalities and maybe new databases are needed. But there are things that become less important, and no one's working in that way anymore. So you can deprecate and not switch off suddenly, but deprecate smoothly and elegantly, and make sure the data is stored, they're available, but maybe a little bit less available, not so searchable. And I think in the GCVR program, one of the things we look at is how you do that lifecycle management. And I think that's a really important thing to do, and it's a responsible thing to do, because otherwise the demand on the people that have to pay for this just grows and grows and grows, and that's not reasonable.
Thanks, Guy.
Yeah, so maybe I can also add, I'm also, I'm not a fan of sort of centralized consolidation of all data in one place. So I think I'm agreeing with what everybody's saying. I think there's a risk to that. And, we've seen it in Africa. There's been a lot of, over the decades, of sort of paternalistic messaging that Africans can't look after their own data and samples. But I can assure you that when there's a problem in the lab, or there's a problem with the server system, African scientists are very, very fast to be there, and they're actually best equipped. They know the systems, they know how things work. And I think there's an inherent risk, large biobanks, the kind of mirroring of biobanks you have to do to actually secure those. We saw that in Christchurch when, the systems went down there. I think there's an inherent danger in only centralizing. And then we also have the issue of sovereignty of data that can also be a problem for consolidating data in one database. So I actually think data standards, sort of structures for holding data that are harmonized, that are standardized, it's a better place to sink our money. We're actually safer with a lot of databases around the world that can talk to each other. The data can talk to each other if we need it to, but that we have regional and national care of those data. And we're seeing it more and more. We're seeing AGARI at Africa CDC, where there's data hosting and much more of this localized storage of data. And I think it's a safer approach, actually. Yeah, thanks.
Any more questions? There we go.
Okay, so I'd like to ask Professor Nikki. Thank you very much for your talk. So the system for data sharing license is very useful for the people trying to share the data. And so also Creative Commons are very widely used. So do you think, is there any legal compatibility of the legal code of the data sharing license to the Creative Commons, depending on some conditions? Thank you.
Yeah, thanks. Great question. So we have tried to use Creative Commons for data sharing in the past, and we've just found that it didn't have quite a bit of the stuff we were after, like benefit sharing as a prime example. Of course, before I started building the data sharing license, I had a conversation with the legal people at Creative Commons, because I also didn't want to encroach on something if they felt that it was their domain. And they felt also said that it was a good parallel or a complementary product, as it were. So we did have all those conversations. Personally, I don't find what I need in Creative Commons for things like data sets. To have an embargo period as an example, benefit sharing as another example, publication of aggregate data, which we use in the health data fields. You're allowed to use the data for aggregate reporting, but not for some detailed risk factor analysis. Those kind of nuances, they're not there in Creative Commons. So those are the kind of nuances that we've really done a lot of work to capture in the data sharing license. Data sharing license expiry, right? Creative Commons doesn't expire. But I think if you have a data sharing agreement with anyone, it's quite common to have an expiry date or a delete by, etc. So to have that option, it's quite important for data sharing. So yeah, I think we are much more tailored to data sets than Creative Commons is able to be. But I think Creative Commons is fantastic. I mean, I just think that there's a kind of niche that it's not able to fulfill. And these are the very nuances that people are anxious about and organizations are anxious about when their data go out there. I know from the health sector, we're happy to share certain types of data in certain formats for certain uses. It's not a blanket okay for everything. And I'm guessing that the same goes for many other data types as well. The same in human genomics, actually. Yeah, what you can do with those data, it's limited. So it's nice to be able to be more nuanced in the T's and C's. Yeah, thanks.
Thanks, Nikki. Another question?
Well, I have a comment regarding the question right now. Well, CC, Creative Commons licenses are based on intellectual property rights. So its intention is to streamline what kind of rights are possessed or, ignored through the licensing terms. But in your data sharing license, do you explicitly say that this license is for natural data or man-made data? Such distinction is made?
It's intended for any data, actually, of any sort. There's really no limit. If you're holding a data set of any form and not just restricted to sciences, biosciences, any sort of data. I mean, it could even be a mathematical model that you're holding that you want to share, right? It could be anything. It could be code. It could be any kind of data set that you're willing to share. And yeah, it's not a legal instrument. And we've made that clear in our T's and C's when you use the data sharing license. I would call it more of a communication tool and an accountability tool. You know, people say, oh, well, how are you going to enforce things? I mean, we're not going to enforce things, but I would argue it's a lot easier to enforce that someone misused the data if you've given them a license that very clearly says you're not allowed to do X, Y, Z with the data. If someone then goes ahead and does that, you've got a very clear case and accountability becomes easier. And I would also say a lot of the misuse that I see in the data world, it's by accident. It's misunderstanding. People didn't mean to do the wrong thing. They just didn't know it's not allowed. And I think trying to close that gap is very helpful. Yeah. Thanks. But great question on the IP. Thank you. Yeah.
Thanks, Nikki. Guy wants to follow up.
I just wanted to highlight this notion of the temporal nature of licenses, which isn't covered by the original CC packages, and I think that's really important because often the need or the openness is most, or open data is more of a challenge at the very beginning of something than it is later on. And so, you know, if you want, you can think about protecting the rights of the scientists in the lab who want the opportunity to publish on their data. So you maybe have more restriction that after a year or so, when they've had a chance to do the work, things will become more open. And you think also in the infectious disease area, where there is a definite commercial window of opportunity that's very early on. So when some major outbreak is happening, then the companies that want to produce vaccines and diagnostics, they want to get in really early, and so if you have restrictions and obligations and things in that early phase, then perhaps when the commercial window is closed, there'll be people are much more comfortable to release data more openly for open research questions. So I think playing with that is a really important part of this discussion about how open is and how we do things in this new world.
So I just want to say very quickly, there's an option to put an embargo in your license. So there's an embargo period option to answer that very question. Yeah.
Thanks so much. We'll take one more question.
Thank you. I suppose that perhaps I understand we've been talking about data sets that can be shared in an open science environment, with licenses for downstream use. But then there's also the added level of complexity and layer of protection for clinical data sets that contain personal health information that are not open, even genomic data that that has personal identifiers, and we have seen the risk that carries being able to give access to these in different environments. You made me think about this, and I wanted to share it if you haven't already heard about this misuse of protected data that was acquired illegally through the regular current regular protocols in the United States, and yet this is with the ABDC project, and yet 20,000 records for children were misused for a race-based type of bogus scientific research, and this was not. I mean, I actually contend the most people use things benevolently. I think that there are bad actors out there, and we have seen this, and so one thing to consider is also that added level of protection, making sure that protocols are in place, not just the licensing, but the data user agreements, IRBs, and other layers to protect how it's shared. Even though we are generating data with funds from the United States government that demand that these data are made available to the community, we still have to put guardrails because it cannot all just be openly available. That wasn't a question, that was more of a response to some of the comments that you shared. But for us to not forget, there are those other added levels of complexity and layers.
Thanks so much. Do you want to respond?
Yeah, I just wanted to agree and just to say in that, in the governance framework, unfortunately, I didn't really have time to go into it, but something like licensing it's just part of the governance framework. And we have, we call them structural and procedural protections, access. It's the physical storing of data that prevents breach. And then it's also the protocols that allow for access. But there's also legislation, there's the ethics of it, which is separate to the equity and the sustainability of all these things. And they're built on SOPs and standards and appropriate practice. So I absolutely agree with what you're saying. And look, I'm a very cynical person, so I was trying to give my positive face to the world. I agree with you, there are many bad actors, and I can assure you as Africans, we see it globally much more than we should. Yeah, thanks.
Thank you so much. So join me in thanking the speakers and the time for the questions. Thanks so much, Ulrike, Nikki, and Guy. And please do engage them a bit further about the tools and, of course, their vast knowledge. So thank you. Oh, a group photo. Where? In front? Thank you. Go the other side.
Vicky, I'm sorry, is it what else?
Yeah, she had to go to.
Okay, good afternoon, everyone. We are going to get started now. So I am Verena Ras. I'll be chairing this next session. It'll be a very short session. We only have 2 talks. Just before we get started, I just wanted to point out that one of our speakers is not able to join us today. This is Dr. Mir Mehrdad Mirsanjani. He's from Iran and so unfortunately cannot make it. In his absence, though, and out of respect for him, I would just like to perhaps ask the delegacy to take the time to read his abstract and to still give him that visibility that he would have had if he had been able to join us. So disappointed that he can't be here. I was personally very much looking forward to his talk, so I'm disappointed to be missing it. But we're just giving him a shout out in his absence. So without further ado, I just want to take a quick second to thank the organizers as well as the steering committee for allowing us to have this biodiversity and environmental climate science track. I do hope it becomes a more regular feature at these conferences, and I do hope that we attract more and more of the biodiversity, climate sciences, and environmental community to this conference, because I do see a lot of parallels, a lot of overlap between the communities. We often are not speaking to each other, and I think this is a perfect platform for us to be able to do that. I myself am a trained conservationist, so I did my PhD in marine biology. And so for me personally, it's really it's a wealthy experience for me to learn from everybody, but to also see those points of interconnection and intersection between the communities. And so, yeah, I wanna challenge the organizers of the next conference to maintain the track. And yeah, for us to, as a community, do a little bit more outreach to that community to get them here. So I will now be calling up our first speaker. This is Prof. Saurabh Raghuvanshi. She's from the University of Delhi in India, and she's going to be giving us a wonderful presentation on microRNA in plants. Sorry,
Thank you. Great. So a very good afternoon to everyone. Well, it's a pleasure to be here. I would start by thanking the organizers for making this visit possible for me. It's a long distance from India, and this is a wonderful place to have a meeting, totally enjoying it. Now, today I choose to speak on microRNA genes, which are basically tiny regulators. And I also take let me first introduce myself. So I come from an institute which is more than 100 years old. It's one of the largest central universities in India. Interestingly, this year, in a single day, we awarded 120,000 degrees in a single day. So it's a mammoth of a institute. I'm proud to be part of it. Now, we basically work on rice for a very long time, and I've been connected to rice since the Rice Genome Sequencing Project. And we have been working a lot on regulatory pathways which are involved in how rice responds to different abiotic stress conditions. And for the past decade, or probably more than that, we have a lot of interest on microRNA genes because they are coming out to be very, very heavily integrated with the regulatory system in all plants, not just rice. And I got encouraged by the discussion that were happening in the past sessions. So I included a couple of slides, sneaked in a couple of slides regarding this data center. And Vedita has already talked about the details of the data center. I'll talk about certain philosophical points about the data center that should be interesting to discuss. Now, microRNAs have been introduced to the forum a couple of times, wonderful talks. So what I would like to say is that in this lecture, we need to concentrate on the fact of this hairpin, these are basically simple RNA Pol2 transcribed genes, which are able to make a hairpin, and then they are processed to make this microRNA, 20 to 24 nucleotide microRNA genes. The science on microRNA genes mostly start from these small mature microRNA sequences, and then we extrapolate the information of this secondary structure over here. So what we want to do here is that we have been actually studying a lot on the regulatory aspect of individual microRNA family, how they are
regulating stress responses, how they are themselves regulated by different calcium signaling pathways. But the point which I want to make today is about the complexities of the microRNA annotation. Now, this became evident to us when we wanted to go from beyond a single family kind of a thing, but to a large, complete repertoire of microRNA population in rice. And then we wanted to move in different species of rice and see the evolution of that function. So we started to figure out what are the total families, and we were actually surprised by this. So there are 4 major databases. One, some of them specific to rice, others are generic. But what is interesting is that the number of microRNA genes for rice is very, very variable. Not just a little bit, but by hundreds. So the point is that why is that happening? And with this kind of a data, we cannot actually go into any kind of an evolutionary study if you don't know the family structure. So we went ahead and we decided that we would try to curate and annotate 2 aspects of the microRNAs. That is, one is the microRNA family structure, and second is the microRNA target network, because that is where the functional evolution happens. So that's what I'll talk about today. It's a 2-step process that we took to figure out which are the valid microRNA families in rice. The first is because there are so many databases talking about so many microRNA genes, so we thought, let us cluster the data and try to find out unique microRNA genes, and then we can extrapolate the data, where are these families. So that's the step 1. Step 2, we plan to do a 2-step validation of the validity of the microRNA genes. Because what happens is the microRNAs are often confused with either siRNA or tacrNA. There are a whole bunch of small RNA population which is there. So we adapted a 2-step validation. One is whether we are able to detect expression. It may sound logical that, of course, you should be able to, but actually that doesn't happen. And the second is whether we are able to detect valid targets for these. And to do that, we downloaded and analyzed about 100 small RNA libraries and 60 degradome libraries. Degradome, for the basic idea, these are 5' RLM race libraries. These are basically NGS-based data which maps the cut site of where the microRNA has actually targeted the gene. So this is kind of a validated microRNA target association. Now, interesting thing comes up when we did this clustering, and then we found that there are a lot of microRNA genes which are shared by multiple databases. However, there are a significant number of database-specific genes. That means none of the databases have the complete repertoire of genes, and we need to actually collate this information at some point of time. And that point of time is now for us, because we need to figure it out. So that's one thing, that every database has a unique microRNA gene family set. Now, when we go ahead and we apply the 2-point validation of microRNA expression and the status of the target, we divide it into these 4 quadrants. So the first one, that's high expression and significant target, we are able to find 684, which is nice. If we subdivide these 2 quadrants into further, we are able to divide it into low expression and no expression ones. What is interesting is that in this low expression one also we are able to find significant target. That means inherently, which is expected also, we have microRNA genes which are actually inherently expressing at a very low level, but they are all biologically active. What is most interesting is this quadrant where the expression level of the microRNA is very high. That means it's a valid phenomena, but we are not able to figure out any targeting of that. Now, this could be several things actually, and these are the models, these are the microRNA models which actually tells us that the targeting of the microRNA to the protein target is not straightforward. There are a lot of transacting factors or intermediary factors which are there, and we need to work further on these very interesting molecules where the expression levels are pretty significant, but we are not able to catch. One thing is that the degradome libraries are not enough, but since we have 60 degradome libraries, it's not that we don't have that much data, But yes, some of them, we would be able to classify them as targets if we increase the degradome libraries, but I'm suspecting many of them will still remain unclassified, that what are they doing if they are not targeting. Is it translational repression? Because that's not known to be very, very prevalent in plants. It's very much there in animals. So that's a open question, but it's all very interesting. Now let's look at the family structure. Now that we have kind of a unified, non-redundant set of a microRNA mature families, let us look how the family structure look like. This is a typical description of a microRNA family, where you have within one family, this is just for the example, you have multiple microRNA genes and spread across the genome. This is incidental that these are all just on chromosome 1, but otherwise they can be spread across the genome. When you align them, you see that the mature region, that's a 2021 nucleotide sequence, is very highly conserved, and there is a lot of variation in the precursor sequences. But within the mature sequence also, you'll find that 1 or 2 bases SNPs are there, and this creates the diversity in the mature sequence. And this diversity has a functional implication because this is the one that is going to identify the target. So second dimension of the complexity is that while we have a particular microRNA family structure, the targets that they aim for, that is something humongous. I'll give you one example of this is MIR 396. It is targeting 133 target genes, and these are all degradome validated target genes. These are not predicted genes. Now, this multiplicity of function is happening because of two molecular phenomena. One, many of these target genes are sharing the exact same microRNA target site. And many of them are sharing the variants of the different microRNA target sequence. So these are two kind of different kind of ways in which microRNAs are expanding their target network. And this is what we wanted to study further. And for this, we changed our perspective and we figured that let us treat the entire microRNA family as an evolutionary unit rather than one different members. So we made a non-redundant, we clubbed in all 888 unique mature sequences into 472 microRNA families. So from now on, I'll be talking about microRNA families. And to figure out the impact of that microRNA family, we defined something known as the impact index. Now, there are two dimensions of this impact index. Impact index in a generic matter would be how much biological impact does a single microRNA family has in the functioning of that organism. And this gets divided into two dimensions. One, the multiplicity of the number of mature microRNA sequences which are coming out within the same family. So multiple microRNA mature sequences. Second is that the function of the targets that they are targeting, the multiplicity of that function or the molecular functions of that, diversity of that molecular function of the targets also define the expanse of the impact of a particular microRNA gene. So these are the two dimensions we actually wanted to figure out. Now, if I want to explain the multiplicity of this valency, why this valency happens, one reason, again, the same slide, because microRNA genes are dispersed across the genome and they will actually have some SNPs within that. So that's one way of figuring out how you can generate multiple mature sequences. Second is, this is interesting. This one is a represented diagram of a hairpin, and this zero position actually represents where the enzyme DCL or dicer makes the cut, and this is the starting point of the mature sequence. Now, when we took in all those small RNA sequences, we figured out that while most of the small RNA tags are getting cut at this precise position, but there are a lot of them which are actually have an offset of 1, 2, 3 bases, either up or downstream. So this creates a heterogeneity in the mature microRNA sequence family coming from the same precursor. It's the same family. Now, what happens is, if there is a different mature that is coming out, is it functionally validated? Is it functional? So interestingly, we found that in some cases, the variant which we identified was the predominant one rather than the one that was actually annotated in the database. So they are functionally validated. And second, very interesting, if you can see this number over here, this is the cut site, 1164. This is 1163. That means if the variant is 1 base pair offset, the cut side by the Argonaut machinery is also offset by 1 base. So they are all functional, and this diversity in the first cut, it can be taken as a kind of a miscut by the dicer, but it's there and it's functional. It doesn't make much effect. It does not change the target so far. It is cutting the same target, but at a slightly different offset site. The third reason for the valency is that there are several microRNA hairpins which are actually processed twice by the DCL machinery. That is absolutely different sequence. That is one sequence coming from here, another coming from here. So these would be totally different sequences. So if we compile all of this, what we see is, and if we try to figure out what are the molecular nature, what are the molecular processes which are actually leading to this kind of a multivalency thing. So interestingly, we find that one of them is gene duplication, and another is the processivity of the DCL1 activity. So gene duplication is providing 1 or 2 base pair changes in the mature sequence, but DCL1 activity is actually providing altogether a different sequence. So this is what creates the complexity of the network. We divide them, all the microRNA families, into this valency from monovalent to hypervalent, and most of them do fall in this monovalent category. But there are certain of them which are very highly high valency. That means from a single microRNA family, you are getting multiple distinct mature sequences. Again, the same thing, if we categorize then both the things, multivalency and the fact that the molecular nature, that is whether it is DCL1 or this is gene duplication, we put them DCL1 dependent ones as category 1 and gene duplication as category 2. And what we find is the red ones actually are the ones which are where the multivalency is coming out simply because of the DCL1 activity. The green ones, the case 2, is because of gene duplication, but there are a whole lot of them where both the events are happening. So when we talk about the complexity of the microRNA function, both the gene duplication as well as the multiple activity of the DCL is actually contributing to this diversity of the microRNA function. Now let's look at the functional diversity of these genes. That is which and how many targets it is targeting. So this is again the number of, the number of target genes which one family can target, and the molecular functions. We choose molecular functions and not biological process because in orthologous conditions, the molecular function is basically kind of a conserved, but genes can acquire a different biological process. Now, we can skip this one. This is just a degrowth sequencing thing. This one is interesting. Most of the analysis actually for when we attach a function to the microRNA genes, we do it with the, I need to stop doing this. We depend on the predicted target genes. So there's a whole lot of algorithms which actually predict the targets of the microRNA genes, but we choose to go a step ahead and we started to compare for every microRNA family network, the predicted targets versus the 5' RLM race validated targets. And we identify a simple percentage matrix over hit. And what hit us really was most of the families, they do not correspond more than 5%. That means if there are 100 targets, just 5% of them are getting validated by RLM race. Now, this is a huge gap in the predicted versus the actual validated ones. It could be the algorithm, but what we also see is that, and what we are also trying to prove here is that this says that the simple presence of a microRNA site in the target gene is not enough to be targeted by that microRNA. There are a lot of factors which are there, which are actually facilitating this process. And this is something that we all should actually, whosoever interested in this, should look at it. And well, this is a pretty diagram, but it doesn't tell you much. Instead, just tells you that how complex things are. But it does tell you one thing, which is that we do find some networks which are non-sativa specific. So that's Oryza sativa and then Oryza rufipogon, things like that. which are not the cultivated ones, but there are microRNA networks which are actually specific to those. And why we are very excited about this is that the data in sativa is very huge. The data in other species of rice is not that much. So if we are not able to find those networks in sativa, then I'm very excited about it, because it's not the limiting data. It is some evolutionary divergence that happens, and this is something that we are looking at right now. Now, when we divide all different families into the number of targets that they can hit, most of them, monovalent families, most of them are within 1 to 10 targets that they can hit simultaneously. One of them is a champion of 133 targets, which I showed you earlier. So this is a division of microRNA impact that we get over here. And this becomes very interesting. These are the description of, this is the summarization of the molecular function of those targets. You can see over here that, although you may not be able to see this is binding. This is mostly transcription factor or regulatory activities, and then some enzymatic reactions. And you can see at this part of the graph, you can see that they are all clustered together, and these activities are kind of not very generally targeted by the microRNA genes. Those are transport activities and whole lot. So this does not come up if you look at the predicted genes, because then you find everything is getting targeted. But if you talk about only the targeted, only the RLM race validated genes, then this is the thing that comes up. Let's change track here. And I have just 1 minute left, but anyhow. So what I want to talk about is what was the philosophy behind developing this data center. Nivedita has already talked about this, that this is the data center that is taking up data from the entire nation over here. What it signifies here is this debate that is going on between the open access and the closed or restricted access phenomena. And why this is coming is simply because we have moved away from model systems. We have moved into the entire population. We have moved into the entire biodiversity. So this phenomena of open access needs to be reinterpreted. Now, with this thing in mind, how to handle this kind of situation where you would like to have everything in open access, but you need to restrict some access at least for some time. So this is an example at the data center of INDA, that's Indian Nucleotide Data Archive. One portion of this, or one section of this INDA is actually totally in total sync with this INSDC repository. So if anybody in India submits over here, they will automatically be shared with any of these repositories. INDA-CA represents a restricted access thing where you can submit, but it's not going to be shared with international servers. But what is important to understand is that they are all interoperable. There is no difference in the, it's not that they are not connected, and the structuring of the data is totally fine. Now, this is the slide which I want to discuss over here. What is happening is that there are two kinds of capacities which nations are trying to build or should try to build. One is the capacity to actually generate data, and another is to have an algorithmic or the data analytic capacity. Now, when I say data analytic capacity, it does not just restrict to basic NGS data analysis or whatever. It actually goes to AI level and higher dimension of studies. Now, why it is important to understand, because in era of AI, when the data becomes huge, there will be a time when there is no movement of the raw data, but it's the extracted information from that data or basic models from that data will be shared. So the algorithm will keep on recursing over the different data sets. You don't need to move the data. Now here every country needs to find a niche of its own, whether they want to stay as a data generator or they want to be a part of global knowledge discovery phenomena. And every country can put themselves into this. I mean, this is my thought process. What is interesting over here is to define this word sovereignty. It does not mean closed data systems. It simply means that every country is developing the capacity to do things. And the FAIR principles need to be reinterpreted, where the findability and the interoperability of the data should remain a constant. You can play with the access as and when the time comes. So I think I'll stop with this. This is the people who have done the microRNA data work. I'm grateful to the funding by DBT and DST. And this is the team at IBDC. And this is the wonderful sunset at Cape Town. Thank you.
Okay, as with all other sessions, we'll have a quick Q&A at the end. Next, we have Dr. Peter Utz from the Virginia Commonwealth University in the USA, who's going to be telling us about curating species descriptions for 12,000 species of reptiles.
All right. Thank you for the opportunity to speak here. I will talk about something nobody else speaks about, and that is not any of the model organisms or humans or human diversity or diseases. I will try to speak about everything else, basically, namely the rest of nature. But I will focus, of course, on a few selected things, so I don't have to talk about everything. So just as an introduction, I do have a background in molecular biology, and as a grad student, I was actually trying to understand the genes and proteins that are involved in limb development. And I used mouse and mouse embryos for that purpose. And one of the reasons why I was interested in that was I wanted to know how traits are determined by molecular mechanisms. And by traits, I mean specifically like anatomical or morphological traits. So I also had this childhood thing with reptiles. So I wanted to understand why do organisms and reptiles in particular look the way they look like. And so I show a snake here because snakes have evolved from lizards, so they lost their legs. So the question is, if you want to understand how snakes evolved, we have to understand how lizards lost their legs. And so I looked at some of these proteins. So you see the blue protein, which is a formin. It's one of these proteins which is involved in limb development, which is the reason why it was called limb deformity originally. And you see also another protein, which is the one which has a couple of domains. So Alex talked about domains. So, and some of you guys may recognize the SH2 and SH3 domains. And so I found out that some of these domains bound to the protein I was interested in. So to make a long story short, I did find out what my protein of interest was doing, although it was not known at the beginning. But it also took me on a journey to dig into like biodiversity, because at the time, which was in the 90s, there was about 4,000 species of snakes known, but nobody had a list of them. And I was a grad student at EMBL in Heidelberg. And so this was at the time when people set up sequence databases at EMBL. And so I walked over to Ture Etzold, who was one of the people who put these databases online, and asked him, Ture, how can we put up a species database for reptiles? And he said, well, just give me a floppy disk. At that time, there were still floppy disks. He said, give me a floppy disk and I will put it online. I said, okay, well, sure, why not? So I gave him a floppy disk. And so we ended up setting up a database, which was probably one of the first biodiversity databases online at all. And we published this paper in 1996. So you can see the database is now 30 years old. It's probably one of the oldest databases in the biodiversity arena. In any case, I will not talk about the next 20 years, which I actually spent on protein research, although this was my main job description. So I did the reptile stuff on the side. So it was kind of a side project. Although I have to say, like during the first 10, 15 years of the reptile database, nobody really noticed it or took notice of it. And only after about 15 years, people started citing it. So it took a while until people actually started to use it. But shortly after I started the database, I was approached by Frank Bisbee, a guy at the University of Reading in the UK, who set up the Catalogue of Life. And some of you guys have mentioned the Catalogue of Life. It was also mentioned by Guy Cochrane in the GBC talk. And so this is actually a metadatabase of about 150 or 160 species or taxonomic databases. So it's kind of a huge project to catalog all the living species on the planet. And so obviously there's other projects which are going in a similar direction, like the NCBI taxonomy, for example, tries to, of course, catalog all the species, but the NCBI only collects stuff which has sequences available. And so we have maybe about 2 million species total, but the NCBI taxonomy has only maybe 400,000 or 500,000. So it's only 1 in 4 species which really has genetic data available. And just to put this all in context, so we have maybe 10 million species on the planet. Nobody knows exactly, but a bit over 2 million have been described and cataloged in the catalog of life, and about 12,000 of them are reptiles. And again, just to put this into context, so there's about 70,000 vertebrate species, so including all the mammals and birds and all that stuff. And of course, there are databases for all of these as well. I would claim that the reptile database is one of the best ones among these databases. In fact, like just the birds have 3 independent databases. And just like last year, the bird guys actually managed to consolidate their list into like one master list of bird species. And there are about a similar number, like 11,000 bird species. It's similar to the reptile stuff. But you can see also that 97% of all species are not vertebrates. They're just like insects and everything else, So that's one thing we also have to keep in mind. So we also work together with some of these other databases. So here's a paper we published together with the amphibian database, which we celebrated like 25 years of databases just like 5 years ago. This was published like 5 years ago. So this is just an example of our database. So some of the stuff we collect. So we do have pictures of tens of thousands of species. We have all the names and stuff, distribution data. Although the maps have been outsourced to IUCN, International Union for the Conservation of Nature, which is also the red list of threatened species. We have, of course, like the type specimens and all that stuff. We have descriptions of 10,000 species now, like literally text descriptions in the database. And we have about 70,000 literature references. And of course, we have links to other databases and so on. So just to put this again into a bit of context, so we have links to the Catalog of Life, but also to the NCBI taxonomy and citizen science projects like iNaturalist, but also a bunch of other things, like the Red List and Wikipedia and so on. Again, like I mean, each of these links has a story on its own because, for example, the NCBI taxonomy, I mean, of course, they collect sequences, but the problem is once they have a sequence, and then people decide that one species actually is 2 species. So you actually have to split the 2 sequences into 2 different names and 2 different entries, which actually often doesn't happen, okay? You have actually quite a mess of sequences which have the wrong names or which have outdated names or which have no names at all because people submit a sequence to GenBank, which has just a placeholder number. And people never actually add the name to that sequence. So there's a lot of sequences which have just like genus name and XYZ number, blah, blah, blah. So all these things, we are things which we try to sort out as well. All right. So as I said, the amount of literature we try to curate is about right now 70,000 papers, which we have in the database. But this is just the tip of the iceberg. So we estimate there may be like 10 times as many papers on reptiles published. And some people have estimated that the whole biodiversity literature may be on the order of 500 million pages. So, and it's not just like papers which have published as PDFs, it's all this stuff which has published like 200 years ago when these things were described. So you have to deal with like centuries of literature in the past, which adds another level of complexity. In any case, so this effort is, of course, not just like a one-way exercise so that we put the papers and curate them and put them in the database. People actually use the data from the database and cite them in their papers. So we have maybe over 5,000 citations in the literature now as well. How do we do that? So, and this is really an arcane method, which I developed when I was a grad student. So I use mostly FileMaker as a database. And don't worry about all the details here. So this is my internal database. And you see there's a lot of buttons here, which are just basically all of these buttons have scripts and stuff, which help to automate the process of curation. So I mean, I can push buttons to import stuff or push buttons to add a reference to like 50 other species or whatever. So and I know I should in the future I need to change this and I need to have this as a distributed web-based database so other people can curate as well. And we are working on that. So we're working with an organization called Plasi, which have developed tools to automatically import species descriptions from the literature, but it's still a biodiversity-focused project. The other question is, how can we use AI for this kind of stuff? And so, as most other people, we are still at the beginning of all that stuff, but we have done a couple of experiments to use AI for literature curation and analysis as well. But let me just ask this question here. So if you ask any of these LLMs, to ask for certain details about, let's say, snakes, and we just ask the question, can you give me the scale counts for all the 4,000 species of snakes? And so, like snakes have basically three important snake scale counts, like the ventral scales and the dorsal scales and the subcaudal, like three numbers basically. And so if you ask any of these LLMs and you just use the API to go through all these 4,000 species names, this is basically what you get. You get numbers from different LLMs in a completely different way. Each of them maybe has 1,000 or so species covered. And I'm just talking about their training data. I'm not asking them to go out to the web and do web searches and stuff, just like using their training data. That's maybe a couple of months old, so it may be a bit different now, but I think the fundamental problem is still the same. So we do find information for 2,000 species in each of them, but at the same time, 50% of them is only found in one of these LLMs. So now we have to ask, okay, well, if we can of course ask these questions to go out to the web and find that stuff, and that's much better, okay? But that's a different issue. One other thing I wanted to get into before I wrap up is one other thing we can ask these LLMs is to read these descriptions and extract data from them. So, for example, if you take the description, like these are just short diagnoses of 4 different species of snakes. If you ask these different things to extract the data and put it in a table, you will see that it works fairly well, but there's still a lot of mistakes. Like half of these tables, these table columns are actually erroneous. So there's some hallucinated data which is not in the description. So if I just ask, can you make a table from that text and put it into these different boxes? So there's still a lot of these red and blue values which are actually not in the actual description. And so it kind of works, but it's not really good, okay? In other cases, it works better. It depends really on the source you have. Okay, we have described this in some papers, and this was just published after ChatGPT came out, so it's a bit outdated, but it still gets the gist of it. And I wanted to stop here at this point and just thank all the people who have been involved. Now, obviously, there's a million people who submitted data and papers, and we help curate stuff. And of course, there's all these students, which have been who have been involved over the years to do some of that work. And you can find more information on our websites, or just email me, or in the question and answer session. Thank you so much.
Okay, it was a brief session, but are there any questions for the speakers?
I know it's late in the afternoon already, so don't blame me.
Let's wake up. Peter, this is just tickling every awesome arthropod genomics and mannequin birds love that I've had in the past. So forgive me for the naivete, but this is like my past life to do genome databases and curation for these organisms. And so this is more of a what is the ideal scenario question for there's like you said, there's so many databases that have been generating all of this data for several different species, and then there's the Earth Biogenome Project, and then there's I5K, and it's all of these. What I'm struggling with is trying to see how interoperability is hindering discovery for evolutionary biology. Do you have some thoughts on how do you train the community for better practices for standardization of these data, and how do you make better connections so that you can run the span of evolutionary distance in order to be able to make some assertions? Like what's the juice? How do we make it better?
I'm not even sure how to address that question because it addresses a bunch of different issues. But you're right, so one of the challenges we have is that we want to basically describe all life on the planet, like phenomenologically, but at the same time molecularly. So we want to understand like what you talked about. Like we want to know, we have millions of species and each species has thousands of genes and proteins and metabolites and all that stuff. And then we have ecology, which has all these thousands of interactions, A eats B and B eats C, blah, blah, blah. So, it just makes your head explode. And that's why we need all these databases, So nobody was talking about like ecological databases here, which I find striking. But that's the meta level beyond. Some people try to do that. They try to look at like the impact of environmental factors on gene expression and blah, 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 all that stuff. So all that stuff comes together, and so we need all of these resources, and then we need them to talk to each other. And and of course, people are working on that kind of stuff, but it's not a super well sorted out problem, I guess.
So maybe a better question is, should Earth Biogenome Project be issuing some standards that all other databases should be adhering to?
Of course, like as I said, like we try to sequence all of these organisms, including all the reptiles and birds and all that stuff. But then for each sequence, you need species names, or actually names for populations. Like we talked about like the African population in humans and all that stuff. So all this, biology and evolution is not something which is super well defined. We don't have boxes into which we can put species. It's all continua basically, and so that's one of the problems we have to deal with. You want to comment on that as well?
No, that's true actually, and what I see is that the depth in which we used to go for model organisms like Arabidopsis and plants, the similar kind of thing is going to happen for almost every species, because those groups are going to generate that kind of a data. And when we want to understand biology as a complete phenomena, so the study of a single species is not good enough, because the functional diversity and the plasticity, every species is a reflection of one successful biological model. So what I see here is that there is going to be an extensive requirement for data integration. The data generation and analysis is going to be distributed. And in this phenomena, distribution should not end up in breaking the ecosystem. So everybody should need to plug in. So that's why standards, unified curation practices, clear-cut data access policies, I think they are going to be more important than they were ever earlier.
Again, so we need standards at every level. So even if you define species or genes, like if you have one gene is not one gene, because then you have all the homologs and all the other species. And so, the functions may slightly differ in different species, and the regulation may differ, blah, blah, blah. So the standards are great, but again, you have to make sure they don't make your head explode, because, yeah.
Thank you. There's another question over there.
Hey, thank you. So, question for Professor Raghuvanshi. Your INDA slide, you made the point that you can have controlled access and you can have open, and you can still be interoperable. You can use the same system and use the same data, same infrastructure to capture these. I guess I've got two questions. One is a straightforward one, and the second maybe less straightforward. The first one is, what is the scope of what you put into controlled access? I heard you, I think it's human, the Indian Genomes projects, but it's, I think maybe COVID, but are other things in there as well? That's the first question. Then the second question is, that's two polar opposites. Is there a continuum? Is there anything in between? Is there perhaps flux? Do things ever move from one state to the other?
Right. So what in our experience is happening is that the controlled or a restricted access data is I think that phenomena is going to be decided on the data itself rather than we can't say that it's a human data or a plant data or something. There are a lot of human data sets which are there in the open access. But it actually depends on what we are seeing is it depends on the project objectives, that there could be a human data which is there in the open access, there could be human biology data which is there in the closed access. So it actually, the decision is happening at the actual data set and not as a generic phenomena for a kind of a data. That's what we are feeling. Second is that the controlled access phenomena, what I see is not a permanent feature. What I believe is that all data sets actually will cycle through these dimensions of they start from the restricted to controlled, and at some point of time they become open. These are all policy decisions. They are not scientific in nature. These are policy decisions which somehow probably gets affected by administrative or political oversights or things like that. But scientifically, what should happen is that the moment it becomes open access, it should be interoperable. You can't start to make, now the data is open, now we make it start to figure out the metadata. So I'm ready, my thought process is I'm ready for the data to be open at any point of time. It's a policy decision from the funder, from the government, from the researcher who have to take. The data center actually does not take that call. The data center provides the avenue that you have to kind of things. It's up to you how to figure it out. What was the second point, or did I answer that?
I think you answered the transition.
Okay, yeah, transition is the flux is, it is happening. I can't actually define a flux. It happens, and for every data set, that's a different time point when it happens. Yeah.
Thank you. You can go ahead.
Okay, thank you for your interesting talk. My question is, how can biological database be used to monitor and better understand the effect of climate change and other environment pressures on reptile populations?
Let me answer first, and then you can also add on to that. That's a very good question, and so there's multiple answers to that. One is, so there are, I mentioned this one citizen science project, iNaturalist, which is basically a project where everybody can contribute observations of species. Whenever you find a flower or a beetle or whatever in your garden, just take a picture and upload it. And so what these guys already found was, and iNaturalist already has like more than 300 million observations. So that's a database on its own. But what you can do is, since people have started doing that 10 years ago, they realize by now that a lot of species have moved further north with increasing temperatures, for example. And they moved up on mountains, for example. Okay, so as long as there's enough people running around on the planet taking pictures of stuff, you can actually have real-time data on the effect of climate change on these populations. And I think it's one of the best examples of how you can actually track these kind of things. We as scientists, we don't have the capacity to do that. There's just too few scientists in the world to do that. Yeah, so you need basically citizen science projects and everybody to join in. And so that's one way you can do that, yeah.
And on that note, before you ask that question, I do want to say there's an iNaturalist challenge happening for this whole weekend. So if you do happen to wander into our reserves, into any of our parks, and you snap a picture of a species that you can confidently identify, you can upload it to the challenge. So
yeah, great talks. Thank you very much, both of you. I was wondering, because both of you have one kind of similar message, I guess. Is it true that you see much more value in comparative analysis and comparative data hosting than is maybe appreciated by most of people?
You want to start first?
Yeah. So, yeah, I mean, at least what we find very interesting is that when you look at comparative biology in different dimensions, we get to understand biology in a manner in which we were not able to do so if we are looking at a single organism. More simply because now the data is being available for that kind of a study where we have wide data across a single species or multiple species. And we do believe that this will help us in understanding the functional plasticity even within a single species if we actually study that particular function, that particular gene network, that particular gene model across related species or even in unrelated species.
Yeah, let me add one or two sentences because, I mean, as you know, nothing in biology makes sense except in the light of evolution. So we need a comparative approach. This applies to both proteins and genes, but also to species themselves. And so one of the proteins I studied in the course of my career was a protein in E. coli. And it turns out that this protein, and I have this thing with proteins of unknown function, so we studied the protein. So at this time, we found this protein of unknown function in E. coli, and we figured out what it was doing. And it turned out, actually, it's conserved from bacteria to humans. And it had no known function like 20 years ago, you know. And so it was a great example of a case where you have a protein which actually does the same thing in E. coli and in humans. In fact, in humans, it's in mitochondria, which are derived from bacteria. So, I mean, for me, this was one of these eye-opening experiences. I thought, oh, wow, you know, so you study one protein and suddenly it applies to everything in the world, basically, you know, like including plants. There were some mutations in plants found. And I think that's why you need the comparative approach.
Any other questions? There's one more.
Oh, there we go. Dr. Yutz, I'm curious about what you're using for your taxonomic authority. So going family, subfamily, genus, species, and all that. I think you mentioned catalog of life, which, if I'm correct, that's GBI.
Yeah, well,
taxonomy backbone, but yet that gets out of date with what the experts think. How do you deal with this?
Well, ironically, the reptile database has become the authority, so people follow us basically, including NCBI and iNaturalist and whatnot, and of course the Catalog of Life, because they import our data. But we make these taxonomic decisions based on the literature. So we read the papers and we make decisions. We have a scientific advisory board as well, which helps to make contentious decisions. And in taxonomy, there's a lot of them when people cannot agree whether are these two populations different species, or is the same species, or is this one genus or two genera, blah, blah, blah. So we have to follow the literature, and if necessary, we go to experts and our advisory board to make these decisions. But eventually, most people follow what's in our database. And I think that's probably true for a bunch of other databases for other taxonomic groups.
Okay, Professor Sarab would like to continue.
Yeah, just something that struck to me what Dr. Guy asked about the flux of the data movement from the control axis. So related to that, the entry into the control axis is something that is very thought of, it's discussed a lot, and then something is actually put into restricted access. It's not entirely on the submitter that, okay, I don't want my data to be accessible, so I can put it over there. There has to be a clear-cut reasoning why do you want to do that. Your funder has to be on board that they agree that the data is to be restricted, because the funder may want the data to be released and the researcher may think otherwise. So the entry into the restricted access is a debated, is a well thought and well discussed phenomena, and it's not a free, entry is not free for all. And then a unified discussion is done to figure out when it should go to the open access. So I would say the flux is not too much, because, and there are two major data sets over there. One, INSACOG, was earlier, but now it is free. Another was the 10,000 human genome sequence data set from Indian populations. That is still under control access. And the funder has actually, what they have done is they have invited research projects based on that data, and they are evaluating those projects, and then the access will be given to the projects whichever are successful. So that's the model that's being figured out, operational right now.
Okay, any final questions? Okay, then I'd like to thank our speakers and for a very wonderful panel session.
Yeah, photo.
It is also a bit early, but there is a tea break now, if I'm correct, Lyndon.
You understand there as well?
Yeah, we'll make the tea break short, and make it 15 minutes, so we'll meet at 3:15. I just want to invite all the poster presenters to take the time to take down your posters in time for the closing ceremony, and then yeah, please all join us at the 3:15 for the final panel session, the announcement of next year's venue, and just a final thanks to all the conference delegates.
All right, good afternoon, everybody, on the graveyard shift of the conference. All right. So welcome. My name is Dr. Yolanda Julius. I'm based at the University of Cape Town. I'm in the Directorate of Research and Internationalization, and my primary role there is in research and development. So I've had many experience working particularly with early career researchers, and I'm very fortunate and feel very welcome to be facilitating the session today. So just to give you a little bit of info how the session will flow, we will start off with me introducing the session. Then I will hand over to the speakers to introduce themselves. Then we will go into the panel discussion, and then we'll take some engagement from the audience, of course. And then we will wrap up with the final question to our panelist speakers. Okay, so let's get going. And I'm hoping you guys will have many questions, and it sparks a bit of conversation amongst us and the audience members over here. Right. All right, so this session is put together to explore the future of biocuration from the perspective of early career researchers. So it offers the opportunity to envision the future of biocuration, including emerging trends and technologies and novel areas of application, and for cross-domain collaboration. And it's to discuss the building and strengthening of communities of practice by fostering global collaboration and exploring innovative approaches to make biocuration more inclusive, interdisciplinary, and impactful over the next decade. All right, so let's get into it. We'll start over. I'll have a seat over here, and then we will ask the panelist speakers to introduce themselves. So yes, so we can start off maybe with Mr. Kwechi. Kwechi, please introduce yourself.
Okay, thank you. So I'm Ezekiel Kwechi Takam, researcher at the University of Geneva, working on everything related to AI, the evaluation of the ethics of AI, AI safety, and the ethics of, I would say, AI-ready data. But of course, we will have the possibility to talk about it during our presentation or conversation, should I say. Thank you.
Sure, thanks, Kwechi. Then I'm hand over for Temitope to introduce himself.
Hello, good evening. I'm happy to be here this evening. My name is Kolawole Temitope, and I'm from Redimas University, and I'm a postgraduate student under Professor Christine Happy. I'm also affiliated to AgBio, which is a training institute where we train undergraduate students to do projects related to bioinformatics. Thank you.
Thanks, Temitope. And then I'll hand over to Dominique.
Hi, everyone. I'm Dominique Anderson. I'm a senior lecturer at SANBI, so the South African National Bioinformatics Institute here in Cape Town. My sort of main passions are about data management and data quality. So I have a bunch of like spanning projects that sort of like have their tentacles in this kind of thing.
Thanks, Dominique. And then I just want to give apologies for Dr. Alex Bateman. Unfortunately, he cannot take part as he has to make his way to the airport. So I'll hand over for Tony to introduce herself.
Good afternoon, everyone. My name is Tsane Tamuhla, and I also am based at Sandy with Dominic. And I'm part of a group called the Health Data Integration Group. And what we're trying to do is integrate genomic data with routine health data to try and get insights from that. And I'm a postdoc. And just as a disclaimer, I'm not a biocurator. I sit downstream of the process. So I'm going to have a bit of a different perspective to the rest of the panelists.
So let's start off by getting some more background information about our panelists. So I'm going to ask them to elaborate a bit about the experience of or the introduction to this field of biocuration. So while Sony was the last one speaking, you can just give us some background of how you got to be involved in biocuration.
So like I said, I'm not a biocurator. I sit more like at the user end. So I use genomic data and routine health data sets in the work that I do. And in doing that work, it also got me interested, and especially when Lyndon reached out to say, would you like to be part of the panel? When I looked at the topic, like for this year to say from data to discovery, led me to thinking, like, what do we do with the data after we create it? Like, how do we make impact and insights and actually, change the world per se with all these beautiful data sets that are like created. And so in the work that I do, a lot of the time we're thinking about equity, data governance, and how can we use these resources equitably to benefit not just us as researchers, but also the communities from which we get these data from, be it biological, human, or plant, or whatever it is. But how do we make sure that the whole system benefits everyone, not just us as researchers? Yeah.
Right, yes. So Dominique, can you also just give us some background? How did you get into this field?
Totally by accident. So I think that when I'm a molecular biologist by training, so I did my PhD in metagenomics, and then I went into industry for 4 years, and industry taught me the value of like data in terms of that ecosystem, right? Because we tend to sort of neglect it when we're in the research space. We're sort of like, oh, we just want to get our project done. And then I came back into academia where I started working in biobanking, and biobanking really sort of like brought out this thing of like the data quality had to be extra high because, that's the biobank standards, and these are ISO standards, and these are, the biobanks are supplying this quality data and this quality material to researchers. And that's sort of how I got into it. And then with the phage consortium, they were looking for data curators, and they were looking for people to build these metadata standards. And it was just something that sparked massive interest in me in terms of saying, how do I start at the beginning of the pipeline and start building the metadata standards that can actually feed into the laboratory information management systems that I build, and so forth, and such and such and such. So it was really just a rolling train that kind of got me into this here.
Thanks, Dominique. And then let's hear from Kwechi. How did you enter this biocuration field? Because there's no one degree that you can have that you study to get here. So what was your pathway?
Okay, thank you very much. So me as well, I won't have the pretension to define myself as a biocurator, but recently I'm dealing a lot with biocurators. And talking about my journey, typically, as I mentioned during my oral presentation, my background is in theology. So I did my bachelor's in theology at the Protestant University of Central Africa, based in Cameroon, my home country. And usually after the bachelor's degree, we already call ourselves to specialize in a precise field related to theology. So I've decided to specialize myself in ethics because it was for me the best bridge to define a link between my theological background and the analysis of a new social contemporary issue. And then the new social contemporary issue that I've decided to work on was artificial intelligence. So I did my dissertation in the ethics of artificial intelligence during my master's degree as well as my PhD degree at the University of Geneva. So precisely around 2023, 2024, I was accepted as fellow at the Beckman Klein Center for Internet and Society at Harvard Law School. And the idea was precisely there to drive a cohort to reflect on the hot topics related to AI. And one of these topics was what we call synthetic data. So typically nowadays, of course, to train AI models, we need data. But the data available in the cyberspace have already been scrapped by AI model. And now AI model needs new form of data to be trained. And as long as we are no more having human-generated data, now what they are doing is to create artificial data, which is called synthetic data. And in that same period, precisely at Stanford University, there was also this initiative of developing a GPT models, but precisely dedicated to the generation of synthetic genomic data. And it was interesting to have an ethical perspective on that generation of synthetic ethical or synthetic genomic data. And this is how I started all this reflection around the ethics or the definition of standards, policies, when dealing with this form of synthetic genomic data. And when coming back to Geneva with the mentorship of Frédéric Lizasek, we established a project around the curation of synthetic genomic data, but in relation to the potentiality or the possibility to use synthetic genomic data in order to tackle the underrepresentation of African genomic data in global databases used to push forward the discovery. So this is how.
Thanks, Kwesi. You are jumping ahead when we're still going to get to that question about emerging technologies, but thanks for actually introducing it already. So let's quickly hear from Temitope also. How did your pathway start to be in this field of biocuration?
All right, thank you very much for the question. So I come from Nigeria, and one of the things that I notice, or we notice in my the institution I work with, is the gap, the wide gap between undergraduate students and graduates. They lack basic bioinformatics skills. So what we do is to first train them, then give them real life projects. And one of the projects we thought about was AMR. AMR is becoming a thing, antimicrobial resistance. And we thought about it that while I was growing up, we have a lot of herbs. I think that's the first line of treatment for some of us that grew up in Africa. And some of them actually worked very well. So we thought about it that what if those herbs could also have compounds, phytochemicals that could also work for a new drug target for antimicrobial resistance. So we thought about doing a large-scale molecular drug docking for each of those plants to find a new drug target. But the first challenge we came about was there is no centralized database for each of those plants. And that's where we came into Biocuration. So the first task we gave the students was that they have to go, and because one of the speakers here talked about that we have to leverage on indigenous knowledge, and that's what we used. We told them to go to those people that sell the herbs. We tell them to ask their grandmas. So we have that list, and we went into database. So we gave them the task to create that. So I'm the project lead for that particular project, and that's how I became as a data creator.
No, that's great. Very interesting pathways of how you all got into this field. So we want to discuss a little bit about that engagement and visibility and barriers to entry. So my first question would be that there might be some barriers to entry for new folks to the field. And what do you think? What do you think makes it difficult for folks to enter the field or even to expand like their career in the field of biocuration?
I think the first thing is knowledge, not even knowing that there is a field called biocuration, because until I saw the International Biocuration Conference, I never knew what we were doing was biocuration. So if early career researchers don't know that there is this particular field, then how would they become a bioculture? The second one I would think about is a standardized field or a professional field. I would say, okay, this is the pathway to the career to becoming a bioculture, and you'll be able to take. I think maybe I came across one or two courses, but there are not so many courses that are really standardized. They are just fragmented across different databases. That's the barrier can go. Thanks, Simitobi. Anyone else want to expand? What are practical steps you think that can lower those barriers to entry? Sure, go ahead, Monique.
So I think that, like, I agree with definitely with your barriers, with what the barriers are. I mean, I think it's a very, very poorly named career path. I think unless you know, and because it spans so many sub-disciplines, I think it's very, very difficult to have like a particular point of entry to it. I think one way that we can try and like overcome those barriers is to create named roles. Right. So if we say that, yes, this thing exists and this is the name of it and this is the pathway to it, I think that you've got a much better chance of like people who are interested in it thinking of it as an actual real career path. I also think that we have very fragmented training in biocuration. I mean, if you think of how many courses you can find online, right, on let's say whole genome analysis, bioinformatic analysis, and then you try and compare that to the number of courses you'll find on biocuration, it's a fragment, right? And you've got to really, really hunt for those things. So I think that we need to really develop that a lot more and put defined roles to biocuration.
Sure, go ahead. Tony?
And just to add to that, I think that can also be a strength in the sense that as biocurators, you shouldn't box yourself into biocuration as a, we're going to get these data sets and make them look so nice and people can find them and use them and share them and fair and care principles. Because I think, like Dr. Mosego said in his earlier talk, there's a whole value chain, and you as a biocurator, you don't have to be stuck in the technical part of it. I mean, if you look at what Dominic and everybody else has said, how they came into it, there's different parts. There's the governance, there's the ethics, there's like innovation. You can go into industry because you know so much about the data because you're so embedded into it. You know what it can or cannot do. So I think in terms of early career researchers, it's just about like looking at the broader chain of how the data fit, like the whole data cycle, where biocuration fits in, and then how you can also get value, not just being a technical person, but also translating and implementing with the knowledge that you have. So that can also help with broadening career prospects in the field.
Thanks, Tony. I think you guys have touched on like the different stakeholders that are involved in this field of biocuration. So how do you think it can better engage, or what can be done to better engage across those different stakeholders, like the different roles in terms of the scientists, the clinicians, data scientists? What do you think can be done differently to make sure that you actually reach that diversity of role players? Any one of you can go? So there's obviously a lot of stakeholders involved in biocuration because there's no defined career path. So what strategies can you use to better engage different stakeholders in this field? Sony, Okwechi, you can go ahead, and then Sony.
Yeah, perhaps I will start. I mean, it's first of all important for us to also try to define what is a biocurator and to expand the definition or the scope of expression of a biocurator. A biocurator shouldn't only be limited, right, to biomedical data, etc. We also have to take into consideration other form of actors that participate in the structuration of data linked to biology, such as, I don't know, anthropologists, sociologists who are also collecting public data. They have to be considered as biocurator. Sociologists who are also, I would say, exploring, you know, the social records of acceptance of some vaccine or drugs. Those can also be considered as biocurators. And as long as we try to expand progressively, while also keep the rigorous methodology, the scope of expression of biocurators, we can expand the community and then build a certain form of ecosystem, a little bit more broader, more stronger, etc. Yeah. So the expansion of the concept of biocurator.
Sure, go ahead.
And I think just to expand, you know, on what he said, especially as people coming into the field, because if you've already been a biocurator for like maybe 20 years or whatever it is, there's a set idea, there's how you trained and how you got yourself into the field. But I think if you are coming in, the real deal is to unbox yourself, like to get yourself out of the thing that you think you're going to do. I think it's the same thing. I come from a background of bioinformatics. And when you're doing bioinformatics, you also tend to box yourself into I'm doing bioinformatics and I'm going to be doing protein this and this, that, and you don't really see that there's so much more that knowledge. Like it's a way of thinking. And I think as early researchers in biocuration, the way that you think as a biocreator to systematically create these things and to keep them going for a long time, that's a skill that can be applied like in a whole lot of different places, and also to expand the field. And in terms of like stakeholders as well, it's not just treating the data as data. Where do they come from? Is it people, communities? And then that data from those communities, have you seen impact in those communities? Like if there's an indigenous plant that has got some sort of medicinal properties, can people in those communities access that drug, or are they still not being able to access it? If it's like whatever it is, I think we need to look, as researchers, we tend to be focused so much on our careers and what we are doing and the questions that we want to ask, that we forget to look up and think, okay, the questions that we're asking, are they actually impacting the world around us? It's not just about how many papers you can publish or what is the hot topic or what is who is funding what, but we need to be more intentional about what we do with the resources that we have and that we've been privileged to be custodians of and stewards of. Yeah.
So maybe let's go to Dominique. Since we've been talking about the communities of practice, do you think there's something that can be done more of? And of course, maybe us gathering here today is one such strategy. But what can be done more to involve the underrepresented groups in expanding this community of practice?
So this is a very, very interesting question. And I think the thing is that what we need to do is we need to take curation to where the needs are, right? We need to meet people where they are. So instead of expecting them to come to us and be like, oh, hi, curate this data for me or something like that, let's meet them where their needs are. I think that's going to be a very, very important step. And, I don't think that we can just talk about, join Biocuration, join this, it's going to be all fantastic and that sort of stuff. I think you need to take a lot of time to build that community. And especially when it comes to underrepresented groups. Some people don't even know this exists. Some people just think it's like, it just happens. It's just there. It's just this, it's part and parcel of what you do on day to day. They don't seem to realize that it's actually like it's a pathway. It's a lot more there's a lot more to it. I think that we really need to meet people with a bit more empathy. I think we need to really, really increase the amount of training and the amount of concentration we do in biocuration as well. Because if I'm sitting in a group, I'm sitting with you guys here, you all know the value, right? You all sort of appreciate it, you're all in it, right? But if I'm going to go and speak to someone in one of my working groups, for example, who has never ever had to deal with this kind of thing, if I'm not meeting them where they are, and if I'm not sort of meeting them with empathy, then I can't find that one champion. I need one champion. Right? One champion changes a lot of things. And I think we've got to meet people where they are.
Also, I wanted to add to what you said, that we have to make sure that the people, the bio-creators, understand the purpose, the importance of the bio-creation they are doing. It's not just that they are gathering data for gathering's sake. For our sake, we told them that what you are doing could actually cure cancer. What you are doing, people can use it to cure endemic disease in your environment. And even when they were discouraged, because they had a purpose in mind. So biocuration is not just trying to gather data, any data you find, it's purpose-driven, and having that at the back of your mind will give you more interest.
Sure, go ahead.
Yeah, following also all these thoughts, I mean, I'm 100% agree with all my panelists. It's also important to understand that, as my brother mentioned, biocuration is not just the gathering of data entries, etc. It also required from biocurators a deep understanding of the field, the deep understanding of the knowledge. And sometimes I will even mention an experiential understanding of the knowledge. So therefore, if we want to expand the biocuration processes or dynamic to underrepresented community, it's just normal, logical, and coherent to have people from this underrepresented community to provide the experiential knowledge of their community in order to structure their data and to curate their data. So for me, it's just a logic to, to integrate and to involve people of underrepresented community in this collective process of co-building data infrastructure, etc.
Sure, go ahead.
And just when we talk about community engagement, I think we really need to move away from focus groups and bringing in people into a room and then asking, we have this great thing that we want to share with you. What do you think? I think we need to take it a step way back when we're conceptualizing something to actually take it to people and say, will it be of value to you? Because we're going to go into those communities, we're going to ask for DNA samples, we're going to ask for plant extracts, we're going to ask for demographic information or anthropological information. But yet we did not ask them if that information is actually going to be of value to them, or we're asking questions that are interesting to us, but not necessarily thinking about the benefit to those communities. So if we can think about when we're collecting the data, if we're going to get, I think I will keep referring back to Dr. Mataro's talk because he really covered like a lot of the stuff here, that communities are the biggest champions of science. And if they can see how what you are doing is adding value, and if they can be a part of it and they can have ownership, then they're more willing to support your initiatives and they're more willing to participate and to also give you insights that you might not have because you have just read papers and come up with the proposal based on what is published in literature, but not necessarily, as my colleague said, what is lived experiences of people. So that's how we can also get communities, not just our professional communities, but just communities all the way to even those people that we get data from. Yeah.
And that is also the importance of spending the scope of expression of who is a biocurator. Therefore, even communities who are fully involved in the process can be also considered a biocurator. And by expanding like that, we also give them the legitimacy to be part of the process, right?
Yes, I agree with you. I think co-creation is very important, not of course just in this field, but I think it has probably been a bit more neglected in biocuration, the co-creation from the beginning. All right, so I want to get into the very touchy subject, of course, the technologies, like the emerging technologies now being used, of course, in the fields of AI, especially like AI and automation. And I know Temitope there will be able to give us some more information. So, so let's discuss the role of these disruptive technologies such as AI and automation in shaping like future practices of biocuration. What are your thoughts on that?
Okay, yeah, thank you very much for the question. So since I've come here since I've listed a lot of tools and I almost get lost in the number of tools that have been listed. And in my mind, I was like, can we just have a pipeline that works best? So in other fields, there are automation going on where you can just sit down and connect your weather app to another app, and it works and it makes decision and it works in agentic AI. And I asked Dr. Elliot, I don't know if he's here, a question when he was giving his workshop on GWAS that can all this pipeline be completed completely by agentic AI? And according to him, he said not yet. He doesn't believe it can. But personally, I believe based on what the applications in other areas that it can be completed, a biocuration process from the beginning, summarizing papers, annotating to the end can be completed. So it's just about putting all these tools together in a pipeline, and maybe after several benchmarking, after several testing, but that does not mean that there will still be a biocurator trying to check, but it will save a lot of time. So I believe biocuration can be automated completely with the help of a biocurator.
So yes, I would also want to hear Kwechi's take on this, since he's also been heavily involved seemingly with the AI and automation.
We're definitely going to be very sad for me to admit that, but I think biocuration will definitely going to be fully automated in the years to come. I initially mentioned that from a theological background, right? Of course, I also did some technical training because it was important for me to have a deep understanding of the object I'm reflecting on. But let me tell you, I was able to biocurate African-related data coming from global repositories without having any form of knowledge on the data I was working on, right? And I was able to do that in less than 2 hours. Of course, I was also collaborating with a mentor who was supporting everything, etc. But this is just to say that we are arriving at the level of technologies where agentic AI are nowadays able, just based on a simple prompt, a clear prompt, a structured prompt, to do what biocurators are nowadays doing. Hence the importance of redefining what is precisely the identity of a biocurator in this AI age. And that's, I mean, I don't have the answer to that question, but it will be fully important for us to already reflect on that and to develop new form of skills, which are, in my modest points of view, the importance of AI literacy. Each biocurator should have an AI literacy, definitely.
Tony, yes, please go ahead.
So I agree with what you're saying to the point. I hope that it doesn't take over curation completely. But I think that the reality is that we're always going to sit with this thing of junk in is junk out. Okay, it does not matter how amazing AI systems are. If we're feeding it junk, it's going to give us junk, right? So the role of human in the loop is still there, I think, and it's going to be there. And AI is going to look at it's going to sort of take away a lot of the mundane tasks, a lot of the sort of the tasks that take time and that are just sort of like repetitive and low energy tasks. And it's going to give us a lot more time to focus on very, very high value outputs. And if we feed those high value outputs back into AI, we're hopefully going to be improving those models so that like it can eventually like build to a point where, you know, we're not just looking at hallucinations and we're not just looking at, you know, I honestly believe that AI will augment our roles. I don't know if it will take over our roles entirely. I hope it doesn't. But yeah, I just think that like that right now AI is the triage in biocuration. But there's still a role for us.
Thanks. So I think while we're on the technologies, the next question is touching on digital platforms and open collaboration. So in your opinion, how does open source tools and shared resources and even virtual communities strengthen the global biocuration networks, particularly in the under-resourced settings?
Okay, well, from someone coming from low resource settings, we depend a lot on open source tools, thanks to them, because with the exchange rate and some other things, most of us are not able to afford resources that are not open source. So if biocuration is going to thrive in low resource settings, definitely there must be open source tools. Then in terms of stakeholders and collaborations, majority of those that are doing well in Africa have other collaborations that are helping them. So I believe that whether we have the resources, we have the talent, we have people who can work, but also we need the infrastructure and also the training, because for me personally, I've learned a lot here that I'm going to also teach to the students. So it's going to be more of, we have senior scientists here, and when during the presentation, I try to think that these people are growing old, they are going out of the field. How much of this knowledge are we able to get from them so that we are able to pass down to other young people too? So there must be networks of getting from senior researchers to early career, then those that are coming.
Sure, go ahead, Dominic.
So I think that having access to digital platforms that are open source bridges an equality gap. So in other words, what we're doing is we're saying that, you know, it's not that, you know, this person or this group or this community can't have access to this kind of thing. They can't learn, they can't, you know, strengthen themselves. They can't like teach themselves. You know what I mean? It's if we don't have these kinds of things, we're actually creating far more inequality. I think about the fact that when we developed our limb system, you know, a commercial limb system will set back a biobank $100,000 per year. Now, I mean, you can't tell me that someone in a low resource setting can't have a biobank because they can't afford that system. So what do? You bridge that gap with open source. You provide that ability. You don't create another barrier for individuals. So I think that's where open source is important.
Shirley, do you have a comment on that? You? No. All right, but while we're on that topic, actually, it speaks also a little bit to equitable partnerships because, of course, that is, of course, a big issue, especially in under-resourced settings. So what do you think actions that the community can take to ensure that the partnerships remain equitable for all parties?
Well, that's like, it's like got a very passionate. I think I've been saying this all the time. I think part of making sure that research is equitable, right? And in speaking to open access is make sure that people know if we have like genotypes that are available and that are out there, but nobody knows that they're out there, then the people that gave those data are not going to benefit from them. So I think we've got very good resources out there, but we are not doing enough to make them visible. Like you'll be so surprised. I was talking to you about your Afro phyto, and I asked you where is it? Like how do I find it? Where do we find it? How do we? I think the challenge is how do we make these data visible so that they can actually be used, and they can be used onward, and they can be used to do good instead of like having these resources there but they're not actually like doing good. And just to go on to like with the AI and all this training stuff, it's also now a question of which data sets or whose data is being used to train these models. When the models are trained, are those people getting the benefits of these things? Do they even have access to GPT that they can also search and find things? Or is it that their data got into that, trained these models, but they will never actually have access to these things? So I think in as much as we're developing tools, we're doing things, we also need to think back to say the communities, the people beyond us as researchers and the immediate people there, are these benefits trickling back to them? Because ultimately, when we went out and said we were doing this research on this topic, this is what we've got these things about how we're going to benefit them, but nobody ever goes back and actually says, was the benefit realized? And I think if we're going to get communities to buy into anything that we're doing and to continue to trust us with their data, then we need to go back and show them this is what your data is doing, can do, and is going to continue to do in the future.
Great. So let's also talk about influence and driving change processes. So what do you think early career researchers can do to drive changes, for example, in influencing data standards or ontologies and ensure fair practices? I mean, fair as in findable, accessible, interoperable.
The first thing I think is volunteering for some of these roles. For instance, I heard about Fair Sharing Champion, whereas you can contribute to curate data, and indirectly you are also contributing. So I think the first thing is volunteering, then also mentoring others, because there are people, even fellow early career researchers, who do not know about biocuration, who do not know that the things that they are doing actually biocuration, or have the knowledge, the good knowledge, the sanitation methods, and some other things that are necessary to enhance their work. So the second thing is trying to pass the knowledge down to others.
And that also goes with the level of visibility I would say we showcase or we highlight towards the work of biocurators. Nowadays, I will talk about the fields I'm most familiar with. Nowadays, most of the groundbreaking AI model applied to biomedical researches are relying on data curated by people, by biocurators. But when you take the paper, scientific paper, of course, highlighting these discoveries, really you will see in the names of authors the mention of biocurators. Really in the paper, even the methodological section, you will see the process, the metric used to curate the data that were exploited by the AI models to be trained. And I think we should also start by highlighting the precise work of biocurators in our papers, in our works, in our scientific discoveries and reliance, definitely.
I also wanted to add that I think bulk creators, especially early careers, that we are looking for an average early career is looking to grow the career. So we might not have enough time to, except if you are a full-time bulk creator. But for some of us, it's because of the projects we are doing, we have to create. So is there a way you can reward early career bulk creators or bulk creators generally, such that it also adds to their career growth?
That actually brings us to another important topic that a few of you have mentioned before, but I think we can unpack it a little bit, and that is about training and the role of training in shaping the biocuration folks and roles of the next era. So there's different models, of course, in training, for example, hackathons, community annotations. What do you think would works best, or what in your experience works best to really build training on and build skills among the early career researchers in terms of training them in biocuration? I think
I'll try to answer this one. So I think that we need to have like models of training that span biocuration. So we need to talk about training that is from entry level all the way through to like more sort of expertise side of things. I think you need to have such a varied scope of training that a person can enter here and they can move along that pathway. I think that a lot of the training is dedicated towards either very, very entry level or very, very specialized, and there's very little that's happening in between. And I think that biocuration, biocuration is like saying bioinformatics, right? It's so broad. Like, what is a biocurator? Can anybody give me like a proper one thing definition of exactly what a biocurator is? Like I build metadata standards. Does that make me a biocurator, or does that make me a standards developer? I think there's so much scope in here, and I think we've got to like try and make more specialized training so that somebody who's interested in, say, for example, the very technical side knows exactly where to go to find technical training. So I think there's got to be like a much broader scope, not just in terms of more volume of training materials and training opportunities, but just like bringing that scope, talking about that scope.
I think also like touching on what Dominic said, maybe it might be worth it if, and I think somebody else mentioned it earlier, to actually within biocuration, as was done within bioinformatics, to have specialties, right? So if there's like the very technical people who are doing, I don't know what they do, they're annotating things, then those can be maybe formal courses that can be part of like curriculums in undergrad or curriculums and masters or etc. If there's things to do with like ethics and governance and whatever it is, then those can be courses that can be part of the social sciences that deal with that. But like if you are able to take this big thing and then identify the leg and the trunk and the whatever of the elephant, then you are with biocuration, we can then take a step back and then have different people fit into different parts of it, instead of somebody saying, because if, like, for example, as a bioinformatics, like if you say I did bioinformatics, everybody's like, oh yeah, so you can code this thing for me. That's like, so I think the same thing with biocuration. When somebody says I'm a biocreator, you're automatically thinking, I am just going to give you my whatever genome sequence, you're going to come and annotate everything, put it out, and then I'm going to have great things going on. So I think we need to go back, take 2-3 steps back, make little categories and then have people slot in, and then we can see where there's already a lot of information and training being done and where the gaps are and how they can be filled. Yeah.
Just in addition with what they said, I think also it shouldn't just stop at training. We have a lot of materials that myself have noted, but some of them just, it's about you watch videos, but I think there should also be the hackathon part of it or real projects that could actually, because there's a difference between you watching videos and doing the real thing itself, and it's in the process that you actually learn manual annotation and some other things. So I think if the ISB, for instance, could organize hackathons, interesting database could come out of it, and in the process, people could get real world life skills that could actually help them.
All right. I think I want to take the opportunity to open the floor a bit for the audience to engage with our panelists. So, are there any questions or comments? Over there, Tony.
Is this working? Yeah. Okay, so I used to be on the ISB committee, and so I can tell you, I just checked, the ISB defines a biocurator. So you can look in there. There was other things that I learned while I was there. So for a while, there was a certificate in biocuration that was being run by the ISB, which was set in Cambridge, but it kind of closed. I think there was not enough people signing up to it. There's a lot of training materials on the ISB website. But I thought the other thing, I mean, I think your discussion has been amazing and I've loved it. But I wanted to point out that a lot of your suggestions, who is going to feed this to the ISB? Because it's great to have this really thought-provoking discussion. But if it just is that, then where are we going? And then the other, so I was thinking, okay, so maybe you need to have funding for biocurators to go to conferences where you think people need to improve their databases or have databases set up and need biocurators. Funding to set up community workshops. So you wanted to go out to the community and you need maybe funding for that. And to give this ownership and value. And I just wanted to say, so the ISB has funding, has micro grants that are $600. They have travel grants. So although people came here on travel grants, there's actually travel grants so that if you wanted to go to a conference that had an area that you were interested in and you wanted to say, I need a voice on this platform, which, I did in an immunology conference. I kind of said to them, hey, you need to have your data. This was 20 years ago, so they weren't really listening. But and then they also do exchange fellowships, which are of over $2,000, so that you can actually have somebody can go to a lab or a group that's really experienced in curation, biocuration, and they can actually spend some time there. So that's like $2,500 available. So yeah, amazing discussion, and just make sure you think about those things and promote that as well. Okay.
Any other comments or questions? Oh, there you go. Sure.
I wanted to officially tell all of you that you're stuck with us. And so I officially dubbed the biocurators. Don't doubt yourselves any longer. The work you have been doing is definitely connected to the work of the society. So just welcome. You've arrived.
Any other comments or questions? The professors in the room. Patty, yes, please go ahead.
I'll take my chances. Hello, Patricia Carvajal here. Just adding a bit to what was being mentioned. Well, I'd like to leave more of an invitation to closely collaborate with the ISCB education committee. It was I was running a workshop on day one, and to my surprise, nobody knew that there exists a profile that has been officially described within a competency framework, ISCB competency framework for a biocurator. And I was leaving an invitation to work on that profile to extend that and reflect a career development. So there is a lot of information, but I feel it's a bit of there's a bit of isolation. Speaking of communities practice, we need to work together, and there is training materials, for instance, but it's difficult to locate them. And maybe it's time to work together with organizations or institutions like, for instance, EBI, which I work for, but perhaps update, for instance, the collection that there's in e-learning for biocuration, but just examples like that, or work with like ISCB and update all those resources that are announced through the platform. So just it's an open invitation for that.
Thanks, Betty. Any other question, comments of the other early career researchers in the room? Oh, sure, here we go.
Well, I want to ask about the relationship between mother tongue or language and creation. In Asia, there are, of course, many languages, and I'm from Japan, but we have 100 million population in Japan. So basically, education is done in Japanese, but for databases, we use English. DDBJ does not have any Japanese information. It's completely in English. But nowadays, people are more careful about mother tongue. And of course, some information can be written only in mother tongue, for example, local knowledge. So in Africa, I heard that yesterday, a population can be grouped into 5 in the entire continent. But how about the language? Do you have any plan to organize your mother tongue?
So I want to tackle this a little bit because language is quite a passion of mine, actually, and it's all got to do with the equity part as well, right? So we know that science is sort of like majority in English, an English spoken subject, right? But the thing is that if we're talking about the fact that we want to meet people where they are and we want to have that empathy, how can we possibly do that if we have not accounted for the fact that a lot of people do not speak English as their first language, right? So it is actually up to us to, and this when I was in my talk where I spoke about like semantics is not just language, right? It's about the fact that there was a very, very interesting article about the fact that in Zulu, for example, there are no words to describe a scientific jargon, right? So we call something by an English term and we think it's absolutely perfectly normal and, it's so common. But there is no singular word to even describe that in another language, right? So if we want to just simply translate our standards into that language, that might not necessarily be possible. And it is for that reason that we're actually going to have to involve communities even more to be able to say, okay, well, in your language, what is the best way for us to describe this so that it has the right meaning and doesn't carry ambiguity? And I think we have to be very cognizant of that kind of thing. And I mean, in South Africa, how many official languages do we have? Right. So and there's it's not just even different languages, it's different dialects of the same language. So we've really, really, really got to be we've really got to be conscious about this.
I think it's a good question. The genetic diversity that you see in the continent also underlies the linguistic diversity. Over 3,000 languages. I think in Zambia alone, there were 77 languages within one country. And in some countries, they have to use English if it's just to come on themselves. So I think it's a very good question, including how do we bring in content of some of these disappearing languages into the cyberspace. A lot of very good work in natural language processing, I think, in this area is coming to the fore. Thank you.
Yes, I was going to add that's probably where the emerging technologies are going to try and help with the translations, but of course it will need rigorous human input.
Yeah, and absolutely. And talking about new technologies, I also have to highlight, it's important to highlight a progressive work that is being developed by some organization in Africa, precisely the Lacuna Fund, trying to curate African languages in order to train AI models that will be quite effective in the translation of this kind of languages. But of course, we have more than 3,000 languages, and until now, they were just able to curate something like 146. The Lacuna Fund, I think you can do some research on that. But they are doing an amazing work. And of course, they are always open to other contributors as long as it's through an open source approach that we are going to create this form of languages. Thank you.
All right, I think we'll wrap up with a closing question to each of the panelists. What do you think, or what best advice would you give to an early career researcher wanting to enter this field right now? I think we can start with Sandy.
I just think that's been like the theme just for everyone, is just to find mentors, like do your research. And when you find a mentor, because I'm a postdoc, I'm in my second year of postdocing. When you're looking for a mentor, there's this compulsion to find the most popular person or the one with the most publications, but you need to find the person that is the right fit for you. And in as much as you are asking for mentorship, you must also be interviewing this person, because they're also interviewing you to see if you'll be a good mentee for them. Is it worth their time to invest into you? Because a mentor-mentee relationship, there's an investment there with like time and effort and all those things. But find a mentor, because that is a person who will open the doors and the rooms and everything to get you to where you want to be. But also be intentional about what it is that you want to do. You have to ask yourself, where do I see myself? Where do I want to go in the future? Like, am I going into academia? Am I going to be in industry? And with that in mind, then you're going to find the mentor who's going to get you to where you want to go. Are you going to stay on continental Africa, or do you want to then go overseas? Those decisions, you have to make them, and you have to be intentional about them. And if you can do that for yourself, then you'll be able to find the right person who can help you on your path. That has worked for me as a personal lived experience. So yeah, find the right mentor, but not popularity, but by what will work best for you. Yeah.
Dominique, closing tip.
I would definitely say that like every single day is an experience to learn. Don't ever forget that. I mean, how many years in, and every single day I'm learning something new. It does not mean I'm incapable. It just means that I'm actually capable of learning. I think follow what you want to do. Curation, guess what? It can be sexy as hell, because the reason I say that is because the skills are so transferable. I think just go for it. Yeah.
Quechi?
Thank you. So in addition to what have been said, I'll just base myself on my own experience, and I will recommend curiosity and AI literacy. It's definitely the curiosity that moved me from theology to the ethics of AI, from the ethics of AI to the ethics of biocuration. And it's precisely curiosity that was going to expose you to a new field, and precisely to the desire of learning from that new field. And beside that, we are in the age of AI. You have to develop AI literacy. You have to be fully involved in the learning, the desire to learn what we call prompt engineering. It's quite impressive what we can produce by, or should I say, relying on this capability, this skill of connecting and communicating with AI through prompt engineering. Thank
you.
Yes, I'm going to take off from where you stop. We have to learn prompt engineering, but it is very difficult, especially during this age. It is possible for you to start using AI without knowing the fundamentals. And you won't know what to prompt if you don't know the fundamentals. So I will advise that you should skip the temptation or overcome the temptation of going into AI first without learning the fundamentals, taking the appropriate courses, what is metadata standards, what is FAIR principles. After that, you could, when the AI is wrong, what to look at when the AI, when you have your output. So while you are trying to go into AI, as I know everyone, especially in our age, will love to work with AI, but do not skip the fundamentals. Then the second thing is mentorship. Um, Throughout my career, even till this stage, mentorship has helped me a lot. So, but like you said, you have to define what you want from your mentors. You have so many tools these days, and you can get lost. We are in a age where there are abundant information, too many courses, YouTube videos. But with the help of a mentor, he or she will be able to guide you to exact way or exact thing or exact courses you should take at the right time.
Thanks, Amitobi. So let me take the opportunity to thank our panelists. I think we really had some insightful conversation and advice from them today. So a round of applause to our panelist speakers. Thanks, everyone. That's the end of our session.
Okay, thank you, everyone. We'll start with the closing ceremony, and I'd like to thank you for sticking around as well. I know the weather's been looking really good and there's lots to explore in Cape Town, so I'll try not to keep you too long. We'll jump in first with just acknowledgement of the travel fellows. So I'd like to invite Cindy, Victoria, and Peter on the stage, please.
I'll be calling them according to the list. Finally, I get on stage. Going to showcase my skills today. Yes. Good afternoon, ladies and gentlemen, and welcome to the most important session of the afternoon. Before we say goodbye to each other, I am here to just award the travel fellows their certificates from their sponsors. Firstly, I'd like to acknowledge the African Open Science Platform, Roche, International Science Society for Biocuration, and the African Bioinformatics Institute for providing sponsorships for our fellows. So I'll be calling their names and they'll receive their certificates from Victoria. I think Victoria can stand that side so that they come to you that side. Yeah, so we can appear in the camera together. Okay, so I would first like to call Umesh Bhatti. Thank you. And I would like to call Blessing Oselu. I'd like to call Kadri Mara. I'd like to call Martin Muigano. I'd like to call Charlize Ricks. Next is Henry Njoku. Next is Kolaole Temitope. And the last travel fellow is Stephen Adiemo. Okay, and this session will also be awarding top 3 presenters. So the third presenter is Umesh Pati. And the second presenter is Kolaole Temitope. And the top presenter is Blessing Oselu. So my 5 minutes of fame is done. I hope you enjoyed my dancing lessons.
Wow, okay. We are coming to the end. Amazing. It's been a journey. So we, as the representative from the ISB, wanted to do some closing remarks and talk a bit about the travel grants that allow all of these people here to show their work and be awarded. So let's not forget about each other after the conference, okay? Let's stay in touch. You have our social media handles. We are again on Blue Sky, on Mastodon, of course. We are in LinkedIn, and we communicate mostly via Slack. So if you're not part of our channel, please join. You have our email, of course, and our website, biocurator.org, where we post also the updates and everything you need to know about the community. And as thank you, Ruth was saying also, you have all the information about everything I'm going to say, grants, micro grants, and also training materials. So please go there and yeah, join us in our variety of channels. For those of you that are not a member, come join. We have yearly memberships that are the fees depend on the member position and the location, so there are different fees. We also have awards, a career award, so you can nominate yourself or you can nominate a colleague of yours. Even the nominations for early career or advanced career awards are open now, so you can start nominating people right now. The nomination closes on May 15, and the voting is going to be since the 1st of June until the 5th. Also, please consider becoming an executive committee member. It's very cool, as you can see. You do have to be a member to become an executive committee member. So join us as a member, and then you can be part of the executive committee. The time commitment is about 3-4 hours per month. It's a volunteer job, so of course everyone that wants to become or wants to contribute as a member, we welcome all the time that they can put on us in a way. And elections, it's also elected, so the nominations are going to be happening, I think, September, and the elections, the voting is in October. There are 11 of us that are members of the executive committee. As you can see, very nice and friendly people. So come join us. The members have other kinds of benefit as well. We have different kinds of awards and grants. You have the Inclusivity Awards. These are awards like for nursing or childcare, mobility devices or specialist transportations, signers or audio-visual aids. We also have exchange fellowships, as Ruth was saying, to facilitate visiting another lab or organization. It could be up to 2,000 Swiss francs. And then we also have micro grants that are to facilitate local or regional meetings. So you can organize a local meeting near to your workplace. And then we have also the travel grants. These are grants that are not only available for members, so they are open to the whole community. The idea is to support travel and accommodation for the ISB Biocuration conferences. We also provide virtual travel grants to cover the cost of the registration. And as I said, it's open to non-members as well. It's available for all kinds of contributors to the field, so biocuration, software engineers, students are welcome to join. Even as part of the award is 1 year of membership. So if you're not a member, but you want to become a member, this could also be a good way to get there. We are called, of course, allow all kinds of applicants from all the different countries and continents. So don't be afraid to apply for them. Even previous winners can apply, but they have a lower priority. This year we gave 5 travel grants, but unfortunately one of the travel recipients couldn't attend, and we're going to hold the travel award for them for another conference. But we did have 4 amazing travel awardees to attend this meeting, as we were just awarding them. The total awarded this year was 2,700 Swiss francs. And again, to receive a travel grant, you need to have an accepted abstract for a conference, but then you are pretty much golden. You don't need to be an ISB member. We do try to prioritize students, postdocs, applicants from low to middle income countries, and applicants from countries that are suffering from natural and humanitarian disasters. And the awards are paid as reimbursements. Once again, congratulations to the Travel Awards winners. And now, let's talk about what's coming next. We have a variety of other virtual conferences sessions. We have the next one is on May 7. It's about AI in biocuration. It's a workshop. You can go and find more information in our webpage, of course. So I think this is a very hot topic, and we are all very interested, so please come and join us for it. Also, in June 15, we have a virtual poster session. If you presented poster here, you are more than welcome to present the same poster in the virtual poster session. And of course, if you didn't present a poster, but you want to, this could be a good idea and a good way to get it out there. Then in September 14, we have Careers in Biocuration. It's a session where there are going to be different talks and a panel discussion. And in October, as we always do, we'll be having the ISB Annual General Meeting, which is a virtual meeting where we announce the winners for the awards and the next projects, trainings, and sessions that we are going to be doing. And next, the most important thing, the thing that everybody was waiting for, the location of the next international conference is going to be in Mishima City, Japan. So I would like to welcome Mr. Masanori Aita.
Thank you for the introduction. Okay, as introduced, I'm Masanori Arita from DDBJ, Japan, and it's okay. Now this map shows how our location in Japan. You see, here is Tokyo, and Mishima is a small city, just southwest, 100 kilometers southwest of Tokyo. And here, there is Mount Fuji. So from our city, you can see the beautiful view of the mountain. And also nearby, even closer, there is an active volcano called Hakone. So you can see enjoy the nature in our city. And from Tokyo to Mishima, it's only 1 hour, little less than 1 hour by Shinkansen bullet train. So if you land in Tokyo, either Narita Airport or Haneda International Airport, it's only a few hours to our city. Okay, let's see the video of our city. Actually, this movie was created by our city originally, and all the scenes are just nearby in walking distance from the venue. So you can really enjoy that picture and that scenery by yourself next year. Okay, as shown, the host is my institute, National Institute of Genetics, and it's all, of course, located in Mishima City, but a little bit far from the venue. The venue is very close to the station, just a few minutes from the station, and there you have many choices for your accommodation. But our institute is how much? Around 15 minutes by car from the station, so it's a bit far. But anyway, if you're interested, let me know. Okay, our theme, the next year theme is the future of biocuration, AI, and knowledge. And this event is mainly supported by our institute. And it's not only for this society, actually, but it's because our institute supports the event, it's defined as an international symposium of the institute. So I would like to invite, of course, all of you, but in addition, other societies and and all researchers from all over the world. So at least we organize a few international events, including this society meeting. And it is also a 40th anniversary of DDBJ. So the period is June 7 to 12, 5 days, but probably the first day is dedicated to the ceremony of DDBJ 40th. So if you focus on your research or session talks, you can join from Tuesday, but I really recommend you to join from the very first day. Some sessions on the first day may be in Japanese, but we plan to introduce AI translation so that you can understand what's going on. Okay? And because our institute hosts this event, registration fee is free. Free registration. Yeah, although you have to pay for your food. But you can really enjoy nice local foods from Japan. And you need to be careful about the language, as I mentioned in my last question, because this is a very small town of population only 100,000, very, very small city. And most of the people in our city do not speak English, but they're very kind, I guarantee. So you can really enjoy the Japanese culture and hospitality next year. Yeah, this ends my presentation. Thank you for listening.
Okay, I won't keep you long at all. Just have a final few remarks that I would like to make. We started the week with beating of drums, and Vicky explained why we beat the drums. I really want to emphasize the significance and impact of having the ISB conference on the African continent for the first time. Sorry. I think it really shows a commitment by the society to make biological data resources more equitable. And as Dr. Rita said, I think yesterday, and it's been reiterated multiple times throughout the week, that there's a desire from underrepresented countries to become more than just data users, but also data providers. So I really think this is a huge step. Through this conference, we've learned of several emerging African biological data resources. Many of them are still in their infancy, and we're also still very disparate. We didn't even know about each other. So this conference represented a great opportunity for us to learn about each other, become aware of each other, and I think it will serve as a launchpad for us on the continent to start our own community of practice, hopefully through initiatives such as the ABI. But I think it also presented a great opportunity for the international society and the international community to become aware of us. Hopefully, this will signify that we can move forward together, both as a continent, but also as an international community. Since we are in our infancy, we'll look to more mature resources for guidance, but I think we can also offer our unique perspectives, experiences, and expertise to make the international resources also a bit more accessible to African researchers. So I really want to thank the ISB for selecting us to host the conference this year. And I look forward to seeing the biocuration community in Africa grow over the next few years and in the future. So those are all my closing remarks. Oh, actually, I forgot one thing. I have a personal note. I think I've attended several conferences over the past few years, but I think this is one of the conferences where I felt most at home. And it's not just because I was part of the organization of the conference, but it's because in the past few years, I've been quite involved in biocuration, but I never really, like many people have stated, I never really knew of the term biocuration and the society that existed. So I've never experienced a conference where I could relate to so many of the presentations provided and just being truly engaged with the entire conference program. So that was really useful. And yeah, I'm looking forward to participating in the society moving forward as well. So with that, thanking the society, there are also many other thanks that I want to go through. I want to reiterate thanks to all our partners and sponsors for making this event possible. We were fortunate to have sponsorship from many organizations. I think a lot of them saw the value of having this conference in Africa for the first time. So we really appreciate that very much. I'll ask you to hold applause because there's many things to go around. Thank you very much to our three keynote speakers, Dr. Ruth Lovering, Dr. Alan Christoffels, Dr. Tiamo Motseja. We really appreciated your talks. I think you added valuable contribution to the conference, and so too all of our invited speakers. We really appreciate you taking the time out of your busy schedules to join us for the conference and really provide your input. I'd like to thank the Dean of the Faculty of Health Sciences for UCT, all the workshop coordinators, session chairs, oral presenters, poster presenters, panelists, and exhibitors. And also I'd like to thank all of the conference delegates for really engaging with the content of the conference, really making this a productive meeting for everyone. I'd like to thank the hotel and the hotel staff for making the conference possible and addressing our queries and issues as we went along. And a special thanks to Vision Media, the AV team at the back, Stephen and his team. They did a great job. Really appreciate them. I'd once again like to thank the scientific committee members, all those who couldn't join us in person, but also those who could join us in person for engaging with us over the past several months and making this conference happen. Thank you very much. And then finally, I want to give one more big thanks to the local organizing team. Many of us are not, it's not necessarily in our job role to plan this event. But nonetheless, they volunteered so much time and effort into this conference and really supported me throughout the planning of the event. So I'd like to invite them up on the stage and have everyone give them a huge round of applause, please. We're missing Tony. Where's Tony? You can join them on the side, please. I also want to highlight two members who couldn't be here. Vicky had to leave yesterday, so she couldn't be with us for the final ceremony. And also Frederique, Alisa said she's been a great support to us while planning this conference, but she couldn't join the conference in person. But yes, this team has worked really hard, not just this week, but over the past several months. So please give them a huge round of applause. So I think with that, we'll officially close the conference. We started the conference with some drum beats, and I'd like to end it with some drum beats as well. I hope everyone enjoys the rest of their stay here in South Africa and have safe travels back home as well. Thank you.
Thanks for you, my kind.
Okay, we can go.
Thanks, Lyndon.